Deep Knowledge Group, and I will be your moderator for today's talk and discussions. We are joined today by a number of professionals in the cancer research field, including therapeutic cancer vaccine development, cancer diagnostics, and biotech investments. And these will be discussing current opportunities, challenges, major breakthroughs, and forecasts in the fields of therapeutic cancer vaccination. The organizer of this three-day conference is Deep Pharma Intelligence, a specialized analytical agency producing regular analytical reports on major areas of high potential in the pharmaceutical and healthcare industries, maintaining ratings of companies and governments based on their innovation potential and business activity in the biotech space, and providing strategic consulting and services to top-tier clients, including major investment funds and banks, family offices, insurance companies, government organizations, and big pharma companies among them too. The company is a joint venture between the two highly specialized UK-based market intelligence hubs in pharma and biotech. Deep Pharma Intelligence is a joint venture founded by the Pharma division of Deep Knowledge Analytics, a specialized subsidiary of Deep Knowledge Analytics and Biopharma Trend, a specialized analytical portal and media resource dedicated to tracking emerging companies, innovations, investments, and trends in the biomedical fields. This conference also serves as a convenient frame for the official launch of a new open access special case study produced by Deep Pharma Intelligence, named 5 High-Impact Cancer Vaccine Platforms, which is actually now freely available for your review at www.deep-pharma.tech. This new report focuses on a segment of cancer vaccine companies. It delivers advanced market profiling, interactive mind maps, information about companies and investors, technology application use cases, and other actionable information. The current report version reviewed the 5 cancer vaccine platforms, which are Antigen Platform, Tumor-Associated Antigens Platform, Cellular Platform, and Oncolytic Virus platform, and the latest breakthrough and potentially disruptive research strategy, Antigenic Essence platform, which are actively developing their products to enter the market of immunotherapy. As a result, we conducted assessment of more than 245 investors, 150 companies and R&D centers, and a detailed analysis of more than 30 of them in total. So today, we will hear from a number of industry professionals on the matter of cancer vaccine development and related topics to them. This will be followed by a panel discussion that will bring everyone together to explore the topics covered in today's session in a deeper and more dynamic manner. You will also be able to ask questions to the speakers in the chat, and the questions will also be addressed by speakers during the panel discussion as well. So let's run through the speakers. To begin today's talk, Dr. Andrei Bavelo, Director of Deep Pharma Intelligence, who will be presenting trends and leading players in the therapeutic cancer vaccines R&D. Next, we will hear from Natalia Hunchar, Lead Biology Analyst of Deep Pharma Intelligence, who will share with us opportunities and challenges of cancer vaccine platforms and the technology perspective. Following Natalia, we will listen to Dr. Piotr Lochov, CEO and Chief Scientific Officer of BioBohemia Incorporated, with a speech entitled BioBohemia, the New Generation Cancer Vaccines. After Piotr, the floor will be given to Dr. Ernst Gotches, partner at Aglaia Oncology Funds, on the topic of cancer vaccines and insights from an investor and entrepreneur. And then we will hear from Christopher M. Coley, the Marketing Development Manager and Digital Pathology at Panasonic Corporation, with a talk from 1 week to 1 day. Take control of your research by automating tissue microarrays, multiplexing, whole slide scanning, and AI analysis for enhanced bioinformatics. And finally, once again, we'll close off today's sessions with an all-speaker roundtable where you can ask any questions to the speakers. Please do see our contact information and linked to the launch report at Deep Pharma Intelligence. Okay. Right, I think we have quite a few people attending, which is lovely. And I can already see that there's some comments established, which is great. So I think let's get started. We will start with Dr. Bovelo. So yes, I'd like to welcome Dr. Andrei Bovelo, the Director of Deep Pharma Intelligence, who will be presenting trends and leading players in the therapeutic cancer vaccines R&D. Andrei, thank you for joining us. The floor is now yours.
Thank you very much, Maxine. Today I'd like to present you with the trends and leading players in the therapeutic cancer vaccines R&D, and I'd like to highlight some findings that we identified as part of the research in our report 5 high-impact cancer vaccine platforms. As Roxy introduced, the report is available for free now on our website. So first of all, let's start with the introduction of what the cancer is. And in order to understand this system disease, we have to understand that essentially cancer is a loss of normal growth process control in cells. Essentially, normal cells, due to some random mutations, due to some external factors, maybe genetic mutations in the human body, they kind of get into the state where they lose control of the normal cell cycle. So they stop dying away when they finish doing their job, and essentially this leads to uncontrolled progression of those kind of cells, and they grow into tumors, which leads to cancer. Essentially, the cancer is very difficult disease to treat for a number of reasons, and one of them is tumor heterogeneity, which is the idea that the cancer tumor is not a uniform thing, but rather it has a diverse set of cancer cells with quite different properties, and this leads to challenges with the actual treatment, because while the treatment might be effective against one line of cells, it might not be as effective or might be completely useless against the mutated cancer cells. And so the whole therapy might not be very useful and might fail to stop the progression of the disease. And there are many approaches right now to treat the cancer, and the oldest ones include things like chemotherapy, and the more modern approaches rely on trying to fix our immune system, and which constitutes the area of immunotherapy. And one of the hot areas of the immunotherapy is the therapeutic cancer vaccination. This is the quite old idea, but only recently it has grown into a very promising area, which already gives hopes for fighting this deliberating disease. So first of all, let's recall how the vaccination works in general. And again, vaccination is very broadly defined thing, and it is used to protect our bodies from pathogens, typically viruses or bacteria. And the idea is fairly simple in general, but quite complex in details. But the idea is that we use kind of either inactive pathogen or some part of that pathogen, antigen, to kind of train our immune system beforehand, so that next time when it encounters the actual disease, it already knows what to fight, and it can get activated and essentially render the immune response, which would be enough to fight the disease. But in the case of cancer, it's a much more complicated story compared to, let's say, viruses and bacteria. Why? Because cancer is essentially based on our own cells. So the cells of the body. And that's why those cells can get quite substantially more sophisticated in the ways they can kind of trick our immune system and avoid its response. That's why it's so difficult and challenging to develop the cancer vaccines. And again, in order to understand how this technology works, let's recall what is the immune system. We have the innate immunity, which is a non-specific immunity, which just protects our body from essentially anything that goes wrong, or any external factors that enter our bodies. And we have adaptive immune system, which is a more complex part of the immunity, which essentially can memorize the information about previous encounters with diseases and hold this memory about the diseases for months and sometimes even years. And so next time when we get ill, we kind of can easily recover. And one important component of the immune system are the antigen presenting cells, for example, dendritic cells here. And the idea here that these cells are kind of messengers who interact first with the pathogen, with any malignancy, and then they kind of get the information about the antigen on those pathogens, and then it presents those antigens on their surface, and then travels to lymph nodes to kind of instruct the specific immune cells, like for example T cells or B cells, and get them activated and then get them prepared to attack that specific pathogen or malignancy or whatever. And for example, one technology that is now really rising, just to give you a hint how this may be done, the mRNA vaccination works like this. We have healthy cells and we have the cancer cells, which essentially healthy cells which acquired some mutations. It is damages in the DNA, and so we can identify those mutations. And then we can kind of identify this, those mutations that would be most useful to render the immune response. Not every mutation is suitable, but some mutations are kind of suitable to develop the vaccine that would render the most strong response. So these kind of mutations can then be represented in the mRNA molecule. And the mRNA molecule is essentially the molecule which is a messenger, the molecule that instructs our body to produce some proteins or some molecules. And then we can ingest this mRNA with the information about the mutations in the cancerous cells into the body. And then this mRNA will be essentially kind of read by the antigen presenting cells, and the information about these antigens will be presented and then delivered to the lymph node to adequately kind of initiate the cells, the T cells, to fight these cancerous cells. So from that moment, they know what to fight, and they can attack the cancerous cells, and in theory, they can kill and cure cancer. So for now, although probably 30 years of research has been done in this area, it just several vaccines that are available and FDA approved for now. Some of them are vaccines kind of preventive cancer vaccines. Those include vaccination against the human papilloma virus or from hepatitis B. And this is not actually the cancer vaccines, but they are vaccines which help to protect us against the very reasons why cancer might emerge. But this report in my presentation will focus on two other areas, the therapeutic cancer vaccines. And those vaccines are vaccines that are administered when the disease already developed. So they are administered at the actual therapeutics to treat already progressing cancer. Also, there are prophylactic cancer vaccines, but we are in very early days of this field, and the idea here is to give patients something that would probably help the immunity fight the cancer in the very first occurrence, and the patient might not even develop the cancer at all. And we have first of its kind preventive breast cancer study right now just initiated. But from the therapeutic cancer vaccines, we only have basically two examples. This is the Prevanga vaccine, which was FDA approved in 2010 against some form of prostate cancer. And then we have the Oncolytic Virus vaccine, telimigan, which is treating the melanoma. It was approved in 2015. And then the interesting thing that the vaccination against tuberculosis is actually was shown to be the way to treat some bladder cancer. So this is the third example of the existing cancer vaccine. So it's not much, but we can expect really the growth in this area. And so if we see the occurrence of clinical trials ongoing right now, we can see that's pretty much a lot of stuff is going on here, and many types of vaccines are now tested. Most of them are in phase 1 trials as of today, and so this brings us to the idea and expectation that soon we are going to see results, and hopefully we can see some progression in this area. And there is total the scientific interest is growing in this area as well. And we see that it's a lot of research that is being done right now, and we see that the research is growing, and also the funding for this area is growing as well, which we see in these two graphs for the leading companies and for many biotech startups. And this means that biotech investors kind of see the light in the darkness for this area, which is a very good sign. And judging by the analysis of the market and on many companies and use cases and latest breakthroughs, we kind of feel that the area now is on the brink of progress, where it will acquire kind of exponential dynamics, and this will be driven primarily by two factors: but the innovative vaccine platforms, for example, mRNAs on Antigenic Essence, and also bioinformatics tools and artificial intelligence, which are really helpful for modeling these things and for identifying best antigens, etc. So we have profiled the markets. And in this chart, you see the categorization of companies, kind of antigen that is used for the vaccine development. So, and this brings us to the classification of antigens. They fall into two broad groups. One is tumor-specific antigens, and another one is tumor-associated antigens, and everything that falls in between. So the tumor-specific antigens are unique molecules, unique proteins, which are available only on cancerous cells. Whereas the tumor-associated antigens are kind of molecules, most probably proteins, that are available in both normal, healthy cells and cancerous cells. But in cancerous cells, they are, let's say, overexpressed, or the profile of their kind of activity concentration might be different. And so this might be a trigger and to develop the therapy based on this difference. And we have whole cell antigens. Essentially, this is the idea that the whole cell can be used to trigger response with the entirety of molecules, antigens that it has. All these methods have their own strengths and weaknesses, and we will be discussing those in more details today. And also we have categorization across the second very important component of any therapeutic vaccine, which is the delivery system. And so we have really new technologies coming, such as mRNA, DNA vaccines, viral vectors, peptides, and dendritic cells, and even CAR-Ts. So all these kind of delivery machineries, they give hope that we are approaching this convergence of biological technologies, and finally we can have meaningful treatments. So reviewing the landscape of companies, we have identifying kind of players with the mainstream approach, like for example, antigen vaccine developers here on the left, on the right side, and then on the left side we have more innovative approach, kind of the companies that are in early days, or maybe in some test modes, and they have a platform that's promised to disrupt the market or complement the existing mainstream approaches. We have classified some of the companies across their funding capacity, the amount of funding that they raised, and the properties of the vaccination method that they develop. For example, those range from they fall into broadly into three groups: those who have mildly specific vaccines but can address that tumor heterogeneity problem, as I mentioned on the slide 2. We have those with highly specific vaccines, but with limited or absent heterogeneity problem addressed. And we have some quite rare examples, but very promising examples of vaccine platforms which address both tumor heterogeneity and specificity. And also we have classifications across the pipeline maturity. So we have platforms with preclinical proof of concept studies, clinical pipelines in phase 1 and 2, and also late stage clinical candidates. So let's start discussing the key platforms that are now popular and promising in this space. So Antigen Platform, as I already showed you, that neoantigens are unique antigens that are derived from unique mutations in the cancer cells. So those antigens are not present in healthy cells, and so they can be very clearly differentiated. Essentially, the technological process looks like we have a collection of tumor and normal tissues, and then of course we have genome sequencing of both kinds of cells. And by the way, we need here to stress out the importance of things like next generation sequencing, which actually makes it possible to sequence DNA, RNA, and all these molecules to be able to actually do this at all. So then we have, based on the sequences of healthy and cancerous cells, we can find the differences and specific kind of markers of the cancerous cells using bioinformatics software, bioinformatics methods. And artificial intelligence, by the way, is very helpful here. And then we have essentially the ability to develop vaccines that rely on those found differences, on those antigens that we identify as the best candidates to render therapeutic response. Among currently available candidates, we can kind of see that BioNTech and Mrna, for example, have clinical candidates, and then Genosia and some other companies. And so, yes, so these are just examples of what they do, and essentially they all rely on this neoantigen concept, where essentially they kind of try to compare the healthy and cancerous tissues and identify the best antigens to work with and to rely on. The next very important platform is Tumor-Associated Antigen Platform. And again, tumor-associated antigens are those kind of proteins that are abnormally expressed by cancer cells. So we can measure this overexpression, and based on that, we can select those kind of proteins which needs to be targeted by the immune system when they are overexpressed. And that's in the that underpins the whole concept of these drug candidates, for example, by Neximmune and Dentrion. And by the way, Provenge is the first FDA-approved cancer vaccines, and this is exactly the vaccine that is using this this technology, this concept. Here it is. Inovio is another example of the quite known company in this space, and we all hope that they will be able to kind of advance their DNA vaccines quite soon. Another broad platform for cancer vaccine research is Cellular Platform, and the idea here is that whole cells of cancers can be used as the source of training knowledge, so to speak, for the immune system. In this case, basically, the problem with this platform is that the cancer cell contains a lot of material, a lot of different molecules, and sometimes not only this renders unnecessary responses, maybe some unnecessary immune response or side effects. Sometimes it may be just lacking the specificity, or some other problems arise. But still, this is a quite significantly studied platform that is used to develop vaccines. And we have just a couple of candidates for brain and ovarian cancer here, for glioblastoma and other types of cancer. And again, we are still waiting for the latest results to understand the where it's going. Some of the companies active in this specific areas are Evita Biomedical, Heat Biologics, and some other companies. So the other platform is quite innovative approach, which is Antigenic Essence platform. And the idea here is something kind of in between of the platform that I have described earlier. So Antigenic Essence platform is kind of a revitalization of the cellular vaccine platform. So the idea here is that only the surface antigens of the cancerous cell are kind of used to create this the mixture, and so the proteases, for example, proteases are used to kind of cleave these proteins and then prepare a specialized essence that would be very representative of a specific cancer cell. So it undergoes the quality control using mass spectrometry, and some proteomics methods are used to kind of make it into a very representative tool to develop the vaccines. One of the speakers for today will describe this platform in great detail, so I will not be talking too much. But the idea of this Antigenic Essence platform is that it is really the innovation behind the whole cell approach, and it's targeting tumor microenvironment, which is extremely important, and it brings cost and time efficiency. So the expert and the company that is developing this innovative platform is Biobohemia, and to our knowledge, that's the first company of this developing this platform. So we can really expect a great breakthrough if things work out well. Oncolytic Viruses is the final and fifth platform that I would like to briefly touch upon. And the idea here that we can basically exploit the idea that oncolytic viruses have the property to infect the cells and induce the cell lysis. And so we can direct the virus to infect cancer cell specifically without touching other cells. And then by that, we kind of stimulate the immune system to attack only cancer cell, trying to kind of kill that virally infected cells. And so it removes the malignancy. There are some candidates again in the clinical trials by the companies such as Oncolytic, Candle Therapeutics, Genelux, and others. So quite interesting developments here, and I think our guest will a little bit expand today on this concept as well. So bringing this all to close, there are kind of key takeaways here. We have the 5 leading platforms. It's not even leading, it's more of the innovation potential that they bring to hope for the sooner discovery of real candidates. And we have profiled more than 150 companies in this area, and a lot of biotech startups are now emerging to tackle this problem. And so cancer vaccine development is moving into the direction of more specific tumor targeting, and while reducing side effects by novel delivery methods. That's also a very important part of the game, delivery systems. And finally, major attention is paid to Antigen Platform, but it will probably be pushed out by the growing cancer vaccine platforms. For example, we believe Antigenic Essence might be a really interesting, innovative platform to kind of advance the market. So with that, I'd like to conclude this presentation. And you are welcome to explore our other reports. We are very active in the area of artificial intelligence and drug discovery, cell therapies, and some other areas of the pharmaceutical industry. And we are open for strategic collaboration and joint analytics reports. So make sure to reach out to us and follow our company on myself, Biotech Oracle, and good luck.
Great, thank you so much, Andrew. That was really insightful, particularly the landscape overview, which really visualized the landscape at the moment. So thank you very much. And I'm looking forward to getting into that a little bit more later in the Q&A. Just before we move on to the next speaker, if I could just kindly remind all the presenters to please have your presentations open and ready, just to avoid any sort of hiccups and issues with connecting and with sharing screens. But yes, okay, let's move on. So I'd now like to welcome to the floor Natalia Hunchar, the lead biology analyst of Deep Pharma Intelligence, and she will share with us opportunities and challenges of cancer vaccine platforms and the technology perspective. Please, Natalia, welcome. The floor is now yours.
Yes. Thank you, Roxy, for introducing, and Andre for a really comprehensive introduction. Now I'm gonna dig deeper into opportunities and challenges of cancer vaccines. And just to be clear, I'm gonna talk mostly about therapeutic cancer vaccines, those which are directed to kill cancer cells when it's already developed. So, to start on a good note, in the recent peer-reviewed papers, it was published that the use of cancer vaccines can improve the cancer therapy tremendously. So, for example, by using cancer vaccine with already established therapy, the responsiveness and efficacy of therapy increased almost fourfold for patients. However, there are still remaining challenges for cancer vaccines, which stops them to hold the big part of the nowadays cancer therapy market. And one of the main concerns of cancer vaccines is safety reason. So, as we already know, cancer vaccines are designed in a way that they're supposed to teach our own immune system via antigen-presenting cells to attack cancer cells and destroy them. However, it's not always the case, and unfortunately, often this immune response grows drastically too much, and cytotoxic T cells start attacking not only cancer cells but own host healthy cells, which creates inflammation, and sometimes it leads even to the patient death. On the other hand, many tumors are still not responsive to their immunotherapy. Such tumors are called cold tumors, which means that they are not visible for the immune system. And in order to make such tumors responsive, they need to be turned first into hot tumors, and only some approaches are able to do that. As an example of such cold tumors, which are a real challenge nowadays, are some kinds of breast cancer and pancreatic cancer. And of course, last but not the least is price and availability. During the last 5 years, cancer vaccine field moved more into personalized approach, especially with the Antigen Platform. However, this approach is quite expensive and not really available to everyone. That is why it faces lots of problems, even right now on the clinical stages of development. So it's quite questionable whether it's gonna be successful on the market. So to give the overview again of our platforms, as we mentioned, we are gonna cover neoantigens, tumor-associated antigens platform, cellular vaccines, antigenic essence, and oncolytic viruses. These platforms are separated by the principle of the antigens which are contained in these cancer vaccines. Just to be clear, antigen is anything which can create the immune response of the organism. So these 4 platforms are separated by the antigens, and they're not really overlapping, but oncolytic viruses, they can overlap a little bit with the antigens contained in here. However, they have a really different biological mechanism of action. That is why we decided to separate them into the another platform. And these platforms can be delivered in different forms. One of the most popular and which were established first are peptides, proteins, and dendritic cells, which currently have the most clinical candidates in the development. However, for example, neoantigens would be presented mostly as nucleic acids, and both of them have some pros and cons. And I would say that coronavirus pandemic impacted into the development of RNA vaccines against cancer as well, since we see some successful examples of coronavirus mRNA vaccine, and it kind of gave more hope and enhanced the development of mRNA vaccines for cancer as well. So to start with, cellular cancer vaccine platform was established the first in the timeline. It's based on quite a simple principle. So if we are having something in our body, we are just trying to immunize it, and we are showing our immune system this something, and we're creating the immunity against this something. So mostly cellular cancer vaccines are presented as lysates of cancer cells. Consequently, since it's the mixture of destroyed cancer cells, is presented as a wide range of cellular antigens. And it has its pros and cons, and I would say that a huge minus of these cancer vaccines is that they have lots of off-target effects because they contain lots of unspecific antigens, and at the same time, those which are needed, those which are specific, they are diluted tremendously. So far, this platform hasn't succeeded on the market. But efforts are still directed towards increasing immunogenicity and reducing the toxicity of such vaccines. And it can be implemented by different ways. For example, Heat Biologics creates an innovative system of delivery of such whole cell vaccines, and they're trying to not only use these antigens, but also use them as a combination, which is quite common strategy when the vaccines are first unsuccessful, then they are used in combinations with, for instance, checkpoint inhibitors, which are already FDA approved. But we cannot really say that it's a classical cancer vaccine. It's a passive immunization. To move to the next more advanced platform, we're going to talk about tumor-associated antigens, which are, as Andrea already introduced, mostly presented as overexpressed proteins. So it means that the concentration of these proteins in cancer cells would be much higher compared to the healthy cells, or the function of these proteins is quite adverse. So it means that they're more active in cancer cells. Also, it can be differentiation antigens. It means that such proteins are meant to be present only in cells which are in early stage of development. It can be stem cells, it can be early embryonic cells. And if they appear in mature cells, it means that most likely this cell is standing on a way of cancer. And also the third group is cancer testis antigens, which were used in the FDA-approved vaccine Provenge, which had a slight success when it was approved, and it passed the stage of activity needed for FDA approval. However, nowadays it doesn't really show that much activity, and researchers still try to improve it because other types of therapy unfortunately give a higher efficiency for prostate cancer compared to the cancer vaccine Provenge. So efforts are basically directed towards innovative delivery forms, which would allow a more specific and directed delivery of cancer vaccine to the cancer tumor site. Also, scientists are trying to target multiple antigens instead of only few. It has several reasons behind. For example, if we target the set of antigens instead of few, it decreases the probability that tumor would escape the immune response. So it has less ways to mutate in a way that it would become again resistant to the cancer therapy which was developed. And of course, tumor-associated antigens, since they're present both in healthy cells and in cancer cells, it means that they can produce enormous immune response and adverse effects. And that is why efforts are directed towards reducing these effects. And such way of reducing the adverse effects, for example, was implemented by Next Immune System. They basically use tumor-associated antigens, which are loaded into so-called synthetic dendritic cells, which are patented by the company, but basically it's nanoparticles which are meant to deliver these antigens directly to the cancer cells via teaching immune cells. So it means that the adverse effects are meant to be less severe, but still their products are on the first stages of clinical development. And now we are moving to one of the biggest platforms nowadays and one of the most advanced ones, Antigen Platform. So what are basically neoantigens? These are highly specific to cancer antigens, which develop mostly as a result of whether viral infection or some specific gene rearrangement or protein coding sequence mutations. So it means that some changes in the DNA sequence. However, these neoantigens are hard to discover and they cannot be found in all types of cancer. To discover neoantigen, the tumor sample must go through the complex bioinformatic tools, data processing, and DNA sequencing, which requires lots of time and effort, and of course, money. That is why the efforts of this platform is directed towards reducing the cost, automatization of bioinformatics stage, which means that scientists are trying to implement artificial intelligence, deep learning, to make the stage of bioinformatic data processing faster and cheaper, since it basically takes the most time during the development of such neoantigen platform. And also, great hopes are laid on this platform because after the unsuccessful examples of Cellular Vaccine Platform and TAA platform, neoantigens are implementing individualized approach, which means that they're searching for the individualized, personalized antigens for each patient, and they will differ for each patient, which is of course good because it would give higher specificity, but on the other hand, it again requires more costs for the development of such vaccine for each patient. And one of the biggest companies for neoantigen platform is BioNTech. One of the directions of BioNTech is shared neoantigens, which means that they establish vaccine with antigens which are present in multiple patients. So one product can be applied for a group of patients. But the most of the effort is still directed towards individualized neoantigen-specific immunotherapy with private neoantigens, which means that each vaccine would contain up to 20 patient-specific neoantigens, which will be loaded into lipoplex in the form of DNA or RNA in the case of BioNTech, and then injected to the patient. One of the platforms which really differs from the previously described ones, but at the same time gathers lots of pros of previously developed platforms is Antigenic Essence. So just to simplify, if we are looking at the cell antigens, approximately 1% of them would be presented on a cell surface. It means that this 1% would be potentially recognized by the immune system. And since they are recognized by the immune system, it makes sense to create the immune response against these 1%. Meanwhile, another cell antigens which are contained inside the cell, basically 99%, they mostly create noise and almost don't participate in the development of anti-tumor immunity. So Antigenic Essence gathers these 1% of surface presented antigens. Antigenic Essence is meant to be developed against solid tumors or tumor vasculature. And since it contains all the surface antigens, it's a wide range of natural antigens, which means that it's a multiple and comprehensive kind of fingerprint of the cell. And at the same time, they are not synthesized synthetically, like it's mostly done in the case of neoantigens and TAA, but they're natural, so they're cut from the cells themselves. So, Antigenic Essence platform can be targeted against tumor cells in cancer. So the immune system is taught to kill cancer cells themselves. But another approach, which is quite promising, is targeting the microenvironment of tumor. And by microenvironment, I mean the vasculature. So anti-angiogenic essence is directed towards the vessels in the tumor. And as we all understand, the tumor needs some factors, oxygen, nutrition to grow. So it's quite a popular approach, which is still getting not that good response as we would want. It's basically targeting this microenvironment, which is right now more and more popular, and researchers dig more and more into this sphere. So efforts of the Antigenic Essence platform are directed towards universal vaccines, which should implement antigenic approach. But meanwhile, this platform also can be used as a personalized vaccines. And also it's quite a promising tool, since this platform might be used as an upgrade of previously developed vaccines, which went, for example, to the third stage of clinical trials, but failed this last stage because of mostly high toxicity to the healthy cells. So in case of Antigenic Essence, there are great hope and first promising results that it will reduce this unspecific toxicity. And this platform is wholly owned by Biobohemia company. And just to give a quick look and comparison, basically it's just cell lines in case of group vaccine, or primary cell culture in case of individualized vaccine, which are grown. Then the antigens are cut from the surface of the cells. Then the next stage is a quality control by mass spectrometry, after which we are getting the ready product, which would be mostly presented as peptides or dendritic cells, which were taught to present antigens by these peptides. So as you remember, these are the biggest platforms basically of cancer vaccine development. And last but not the least are Oncolytic Viruses platform. They can actually target neoantigens and tumor-associated antigens. But why they are so different? Because it's not a biological molecules, but it's a biological agents. But instead of causing the disease itself and ruining the healthy cells, they're genetically modified in a way to target cancer cells and to induce their lysis. So they basically destroy cancer itself and create a local inflammation in the cancer sites. However, this platform is still facing some challenges. For example, efforts are directed towards reducing undesirable immune response. Since viruses are live biological agents, it's harder to control them, and frequently the usage of these oncolytic viruses causes a too high immune response. At the same time, innovative delivery methods are created because mostly, even nowadays, these oncolytic viruses are injected through the needle into the tumor. But we understand that not all tumors can be treated in such way, like, for example, brain tumors. So innovative delivery methods are created for this instance. And also fighting ethical concerns. Why? Because on the first stage of creating such an oncolytic virus, the genome of the virus is changed. So it might cause some concerns in some countries. It's really specific, but yeah, still facing such challenges. And one of the companies, as an example, is Oncolytic, which is basically showing how oncolytic viruses can turn cold tumors into hot tumors. As we discussed, cold tumors are those which are not visible for the immune system, so they don't have infiltrated lymphocytes. But oncolytic viruses can cause these local tumor inflammation, which means that tumors will be infiltrated by lymphocytes. And since this local inflammation is growing, the antigen presenting will be also growing, and it creates this self-enhancing loop of cancer fighting. So that is why oncolytic viruses are quite promising and a specific platform, and it already has one FDA-approved product, TVAC. And just to sum up, the future directions of cancer vaccines development is, first of all, more targeted approaches to reduce toxicity. So how to do that? Mostly, it's a use of wide set of specific antigens. So now we would mostly not need the vaccines which would use only one neoantigen or only one tumor-associated antigen. And specifically, for example, Antigenic Essence would present the whole range of antigens presented on the cell surface, which is quite corresponding to this strategy. On the other hand, still finding the compromise between personalized and group approach, since we need to find the best solution between efficacy and price and availability ratio. And of course, direction which is developing is immunoprevention and antigenic strategy. So it means that we want not only to destroy the cancer when it's already developed, but to prevent it and not target only cancer cells themselves, but their microenvironment. Because, as it was shown by many researchers, microenvironment plays a tremendous role in the cancer development, and we cannot ignore this part of the cancer development while fighting with this disease. And with this, I want to thank you for your attention, and you're welcome to ask questions.
Thank you very much, Natalia. That was really insightful. And thank you very much for going through the platform from the perspective of technology. Just before we move on, I'd just like to quickly draw your attention down to the chat and to the Q&A. Please do, we encourage you to converse with one another, to comment on what you're hearing and seeing as well. And also please do feel free to pose questions and pop them into the Q&A section, which you'll find at the bottom of the screen. With regards to the Q&A, we will, of course, be hopefully answering some of these questions directly to with the panelists later on. And speakers and panelists are also welcome to answer the Q&As within chat as well. So please do have a look at the Q&A section, and we do invite open conversation and invite you to discuss and feedback everything that you've heard today. So thank you very much again, Natalia. So I think now let's move on. I'd like to invite Dr. Piotr Lochov, the CEO and Chief Scientific Officer of BioBohemia Incorporated, with a speech on BioBohemia, new generation cancer vaccines. Please, Dr. Piotr, I hope I've pronounced that right. If I haven't, please do correct me, and I'll allow you to introduce yourself again.
All right.
Well. Thank you.
Thank you, Roxy. Thank you, Roxy. Okay. Just a moment. Just a moment. Okay. I want to start with many thanks to Deep Pharma Intelligence for this opportunity to present our company, to present our work and share our ideas, share our technology to global audience. And I'd like to start with several words about me. My name is Piotr Lochov. I am Chief Executive Officer of BioBohemia, and at the same time, I am Chief Scientific Officer. I'm a scientist. My general scientific experience almost 25 years. 20 years ago, I got a philosophy degree in biochemistry. Currently, I'm also a head of metabolomics laboratory. I'm co-founder of BioBohemia Incorporated. And several words about BioBohemia. BioBohemia is a preclinical stage company located in the United States, in Boston, in Cambridge. Main technology is Antigenic Essence platform, intended to develop numerous cancer vaccines by using of proteomics technology together with cell culture technology. So, a title of my presentation you can see on the screen: BioBohemia, new generation cancer vaccines. Forward-looking statements. Several seconds. The problem and its relevance. As you know, cancer is a leading cause of death worldwide, and cancer prevalence is only increasing. Approximately from 20 million new cases in 2020 up to 30 million in 2040. The main reason for this is that there are no simultaneously effective and at the same time long-lasting and without sufficient side effects therapy. So vaccination may offer such kind of therapy because one or several injections may induce body defense mechanism, and by this way, body can be protected for a long time. Main principle of vaccination, well-known principle: if you're going to induce immune response against some object, you should use this object for vaccination. And we can see numerous attempts to vaccinate with inactivated cancer cells, and at least 16 cancer cell vaccines were tested in clinical trials, but without any success. Our solution, Antigenic Essence. This is innovative composition for preparing new generation cancer vaccines. Antigenic Essence can be considered as a deep upgrade of conventional cellular cancer vaccines. It has same mechanism of action, but without disadvantages and with sufficient advantages over them. And among these, a high efficiency, safety, pure composition, control composition, and precise targeting of tumor cells, and numerous other benefits. Antigenic essence can be prepared for any type of cancer cells and for any type of cells. And among these endothelial cells, it means that we can target tumor vessels, and by this way we can treat almost all solid tumors, because solid tumors can't grow without feeding. Several words about background. As you know, native antigens provide the most accurate targeting of immune response, and a comprehensive source of such kind of antigens is cells. However, whole cells may contain numerous undesired vaccination antigens. For example, molecules related to essential cellular functions like biosynthesis, energy balance, and cell division are common for all cells in body. It means that such kind molecules are unspecific and undesired for vaccination. And these molecules consist of 99% of cell content. Moreover, this content unavailable for immune system, because immune system act by means of antibodies and cytotoxic lymphocytes. And both of them can penetrate inside cells and interact with cell surface. So only antigens presented on cell surface should be used for vaccination, because these antigens form actual antigenic profile of cells. It doesn't mean that such kind of antigens we can't find inside cells. For example, receptors on the cell surface are synthesized inside cells can act as a target for immune system only when they are presented on cell surface. So if we use only this fraction of antigens, we may vaccinate and induce very precise immune response and undiluted immune response by undesired intracellular content. Our technology. We have very simple technology. If cell surface antigens are available for antibodies as well as available for cytotoxic lymphocytes, they are also available for protease. So we use trypsin as protease, to treat live cells in mild conditions in order to prevent destruction of cell wall and release intracellular content, and by this way to pollute antigenic essence. And by this way we can collect only cell surface fraction, and this fraction is named Antigenic Essence. And we have additional possibility to analyze this composition, because Antigenic Essence is a peptide composition. So we can use peptide mass spectrometry to define a composition to be sure that we have cell surface fraction, and this fraction is not polluted by intercellular content and is not polluted by trypsin itself. So our technology can be considered as a technology to collect cell surface antigens, and antigenic essence is only desired for vaccination antigens of cells, and antigenic essence is only 1% of cell. And we can encode antigenic essence composition and use this code to find optimal compositions for vaccination in research and development. Proof of concept. We have data in vitro as well as in vivo. for in vitro, we have results for cytotoxicity assays, which can be considered as in vitro model for vaccination. And we compared antigenic essence with whole cell lysate. And antigenic essence being only 1% of cell kills target cells even better than whole cell. And in vitro, we confirmed that antigenic essence can induce immune response, and this immune response can prevent tumor growth. There are numerous approaches for vaccination. The main message of this slide that we have main approaches based on, for example, on tumor-associated antigens or on vaccination by whole cells. And we have approaches which can improve base approaches, and by this way, new approaches can appear. For example, CAR-T technology can be considered as an improvement of vaccination by tumor-associated antigens, and we know that this approach succeeded for bloody cancers. We have same situation with vaccination with whole cells. Antigenic essence can be considered as a sufficient to prove it. This main well-known and widespread approach. It takes all benefits from vaccination with whole cells, a diversity of native antigens, and by improving of this approach, we believe that new direction will appear. Just main points related to our technology. Antigenic essence is very safe approach because antigenic essence consists of only 1% of cell content. It's free from undesired for vaccination blood intracellular content. Antigenic essence is a natural and non-toxic product, and we expect that we will see a few side effects for our product. We have a very precise approach because we can use mass spectrometry to analyze the composition of antigenic essence. This is almost impossible for whole cells because if you will try to analyze whole cell, you will find that you can see only big mass spectrometry peaks related to, for example, housekeeping proteins located inside cells, and they are common for all cells. Now we have results related to specificity of our approach, and we confirmed that antigenic essence very specific for each type of cells, very specific. So we have universal approach. Why? Because we can prepare antigenic essence for any type of cells, not only for cancer cells. We can prepare for endothelial cells or any other cells. It means that we can treat any tumor, any solid tumor, and we get any solid tumor by targeting of endothelial cells. Main target, tumor endothelial cells, TECs. Product which target tumor endothelial cells was named Santavac. Santavac is an abbreviation, set of all natural target antigens for vaccination. Tumor antigens, sorry, for vaccination. This is a highly specific, highly efficient approach. Why? Because destruction, a small amount of endothelial cells in tumor vasculature may prevent a tumor growth because vascular net will be destroyed. Tumor can't grow without feeding and without oxygen. This is universal approach. And we expect that the vaccination with this product will be stable because endothelial cells less likely to develop escape mutation than cancer cells. And here you can see a workflow to produce Santavac. We are going to use primary culture of endothelial cells. To do this, we are going to isolate endothelial cells from fat tissue. And then primary culture is established. The cells should be propagated in order to reach required amount of cells. After that, we stimulate these cells by tumor conditioned medium. And conditions were established in our research and development in order to induce tumor-specific phenotype. So after that, we can apply our technology. We treat cells with trypsin, highly purified trypsin, in order to split off cell surface antigens, and after that we can collect cell surface antigens, and we can analyze composition. We can compare the composition of antigenic essence with code of target cells. And by means on mixing with montanide ICA 51, we can produce peptide vaccine, or we can load dendritic cells in order to produce the dendritic cell-based vaccine. Another direction of our efforts is an upgrade of well-known and already tested cancer cell vaccines, which failed in clinical trials. And by this way, we can upgrade almost 16 candidates. To do this, we take cancer cells involved in production of whole cell-based vaccines, and instead of cells, we are going to use antigenic essence of these cells. And by this way, we can get all benefits connected with our technology, and we can produce products protected by our intellectual property. Antigenic essence has all benefits of cancer cells and numerous very sufficient benefits. One of them, we can increase a dose of vaccination. As you know, vaccines demonstrate dose-dependent efficacy, and usually a maximum possible dose is used for vaccination. Usually in clinical trial phase 1, such kind of dose should be established, and maximum dose can't be increased because side effects will be almost unacceptable. We have only 1% of cell content. It means that we can sufficiently increase of dose of vaccination up to 2 order of magnitude, and without exceeding a maximum permissible dose of vaccination. And by this way, we can meet to FDA requirements related to efficacy of vaccines. Additionally, we can encode our composition. This is impossible for whole cells, for original cell vaccines. And we have very safe composition because no cells, no proteins, no viruses, no prions, no supramolecular structures, only peptides. Peptides can be easily purified by ultrafiltration. And at this stage, we can remove sequencing grade trypsin. No ballast content, decreases side effects, and numerous other benefits. And additionally, we can use mass spectrometry, and we are going to use mass spectrometry to confirm a composition, and mass spectrometry is embedded in our quality control program and quality insurance program to authenticate our composition. And maybe you know, authentication of cell cultures is a big challenge. I'm talking about now cancer cell-based vaccines because the cells during culturing tend to be changed. We haven't such kind of problem. And we have highly specific composition to target cells. This test is included in our quality control program. Our IP portfolio, we have many patents. I may say that our technology is protected almost worldwide. Our technology was published in numerous scientific papers in journals related to cancer, to vaccines, and to proteomics, because as you can understand from my presentation, we use proteomics to produce our product. And we have publication in mass spectrometry related journal. Our trademark Santavac protected in numerous countries, and the list of such kind of countries can be increased in any time. Here you can see a list of our candidates. We have 7 candidates. We will start with Santavac, a therapeutic peptide vaccine against renal cell cancer. And we are going to register IND in 2023. And just after that, obtained experience will be used to start all other products, and among these 3 Santavac products. 2 of them relates to preventive Santavac, and 2 relates to dendritic cell-based vaccines. We can vaccinate with 1% of whole cell content. It means that we can meet to FDA requirements regarding to preventive vaccines. Because in case of preventive vaccines, we are going to vaccinate healthy people. That is why vaccines should be very safe with low side effects. So we have 2 types of Santavac, for therapeutic purposes and for preventive purposes. One product is very strong product, and some side effects can be expected. Santavac for preventive purposes, less powerful, but almost it's expected without almost side effects. And additionally, we have 3 products which are upgrade of well-known vaccines. Indications. Santavac should be used to treat renal cell cancer. This is cancer is sensitive to antigenic therapy. That is why this cancer was selected. We're going to succeed with our first product. Also, you can see other indications, enamel in prostate cancer, colorectal cancer, lung cancer, widespread cancers, and we're going to use melanoma. Melanoma is well measurable cancer. That is why melanoma is most suitable for human in first studies. And additionally, we're going to discuss with FDA rare diseases and pediatric cancer. Because FDA has accelerated program for such kind cancers. We have a very large market opportunity, almost $200 billion, and market will increase 34% from 2015 to 2030. We have very good business model. We have diversification of profit making. Our main goal to get approved vaccine or vaccines. And by this way, we may have a long-term revenue. But when our technology will be accepted by science or by tech, we will be ready to sell licenses for using of our technology platform or for products, Antigenic Essence products. Based on information available on the market, we predicted a growth of valuation of our company. We have common stages for startup company which work in same field of biotech, and our valuation value inflection points are usual and connected with approval of IND by FDA, when we get fast track designation, and the valuation will increase at each stage of clinical trials. And as you can realize from this slide, we're going to participate in fundraising just after New Year's holidays. We will participate in seed round. Core team. The message of this slide is that Biomarkers has experienced team. 2 clue persons who participated in development of Antigenic Essence platform, me and my wife Yelena, were included in core team. Our partners, IQVIA, is well known and leading consulting company in biotech. And when we will be ready to produce our products, we are going to test our product in preclinical trials with translational drug development company, and we already have offer from this company. This is a well-known, maybe #1 company in preclinical trials. And now I'd like to finalize my presentation in several words about main points. We have technology, innovative technology. We have very large market opportunity. We have very strong regulatory strategy. Our technology is well protected. We have numerous patents. We have patents in USA, in main countries in Europe, in Asia. So, and our portfolio can be increased. I mean, we're going to get additional patents, and we have experienced team. So, and we have well-recognized partners. Okay, thank you very much for your attention. And if you have questions, I will be ready to answer your questions, or you can write to me. You can see our contact information on the screen. So,
Thank you very much. Thank you, Piotr. That was very, very insightful, and looking forward to really exploring that a little bit further later on in the Q&A. I can already see that we have several really interesting questions and comments as well, which I hope everybody will be participating with later on. And also, just to draw your attention back to chat, you can find all the details of the speakers and the companies, as well as all the social media handles as well, and also our Deep Pharma Intelligence social handles and the reports which were mentioned earlier as well. Okay, so moving on, I'd now like to welcome to the floor Dr. Ernst Gütjes, partner at Agalea Oncology Funds, on the topic of cancer vaccines, and he will be bringing insights from the perspective of an investor and entrepreneur. So welcome to the conference, Ernst, and really looking forward to your perspective.
Yeah, thanks, Roxy. Thank you very much for the kind introduction, and very much looking forward to present the learnings that we've obtained since almost our 70 years involvement in the cancer vaccine space. Let me see. I can put this one online. Right. This is my disclosure slide. So I work for an investment firm that has incubated a cancer vaccine company called ISO Pharmaceuticals. That was already in 2004, amongst other companies that are active in the immuno-oncology space. Now, for the topics of today, we'll be covering four subjects. First, I will briefly discuss the history of the cancer vaccine space. We'll be sharing some considerations with regards to antigen selection, platform selection, clinical strategy, and we'll be sharing some concrete recommendations for entrepreneurs that are active in this space. Now, the cancer vaccine field has existed actually for a long time, as some of the previous speakers already highlighted. And similar to many other innovative drug modalities in the field, the cancer vaccine space also undergoing a typical innovation hype cycle, where peaks of inflated expectations are followed by valleys of disillusionment. So the field kind of started, I would say, with a surgeon called Coley, who found out that patients that presented themselves with sarcomas, he could actually infect those tumors with live bacteria. And he found that some patients actually curiously spontaneous regressions of the tumors. So that was really the first concept of a cancer vaccine. Now later on, the field was a little bit silent. And in the 80s, people started using tumor lysates, so whole extracts from tumor cell lines, but also tumors from patients, which were given to patients either alone or in combination with bacterial adjuvants. There were a number of clinical trials there, but they were largely unsuccessful for reasons mentioned before. So if you have a lysate that contains a lot of different antigens, many of them are self-antigens, are not unique to the tumor. Basically, those are flooding the MHC class molecules that
Dr.
present these
Gütjes,
peptides.
apologies for interrupting you. Would you kindly be able to open your presentation full screen, if you wouldn't mind?
Let me check.
Thank you. Apologies for the interruption.
No worries. Is that working?
No. So I think at the bottom there is the little sort of it's the fourth icon along. If you just click on that, that should allow you to do it. So at the bottom of the screen,
let me see.
So you should have 1, 2, 3, 4 icons, and it looks like a wine cup. I can't think of how to describe it. The icon. If you click on that, that should hopefully open it full screen for you.
Is that working?
No. So if you go to the bottom, yes, you just had your cursor on it there. So sorry, F5, someone kindly
F5.
offered that.
Let me give it a try.
Thank you very much, Vladislav. Thank you. Try F5, that should hopefully do it.
Is that better?
No. So if you go to the bottom of the screen,
yeah,
of your PowerPoint, if you go towards the right to where it's got the zoom and it's currently on 62%, just to the left of that, there is an icon by the minus symbol. If you click on that, if F5 hasn't worked, otherwise we're happy to proceed. It was just in case it was possible to adjust that.
I can't see the icon actually. Strange, because we actually use Zoom in our firm. So, that's strange.
Don't worry, we can proceed, not to worry. Are you on the Zoom screen or are you on your presentation? Have you got PowerPoint open at the moment?
I've got PowerPoint open at the moment, yeah.
So it should be right at the bottom of your screen. So if you take your mouse to the bottom of the PowerPoint, and as if you're about to zoom into, yes. Now, if you go to the left, just to the left, not to the far left, to the left of where you were zooming in, there's an icon there. There's 1, 2, 3, 4 icons there. Yes, left. So if you go to the right now, right again, and again, and again. That should be fine as well. It's I believe it's the one on the end. So it's right again. Yes, that should do it.
Is that one?
No, it's towards the right. Don't worry. I think we should, if we can go back to.
Yeah, let me bring it up again.
Yes, no problem.
Is that the one?
Don't worry, we have time. It's not a problem.
Yeah, no problem.
Yes, that's the one. That should do it for you, hopefully. I believe. That's really strange. So if you want to go to the one on the left, which had the read, the book icon, that might be okay. Otherwise, please proceed as you were.
The read icon.
That's strange that it's not allowing you to go to the screen now.
Well, anyway, I hope you can still see the slides sufficiently.
It's sort of zoomed. We can see 6 of your slides on one page at the moment.
Still?
Click on Vanaf begin. I don't know if that means anything. Somebody has volunteered that in chat. That should be fine. So if you just want to go back to your initial slide, again, apologies for the interruption. That should
no worries.
Or double click on the slide. Apparently somebody said that usually works as well on the panel.
Is this better or still the 6 slides?
No, it's okay. We've got it back to the very original, but it's
okay.
zoomed in slightly. So if you go back to the slide where you were presenting, so I don't know if you're on the second or the third. So if you double click on number 4.
This is number 4.
Yes, if you so on the left, if you double click there.
Yeah.
Otherwise, don't worry. It might just be an issue with or press Control plus Control and F5 together. That might do it as well. We have some IT experts, I think, here in chat.
That's strange. Yeah.
Don't worry. I think that it might just be something that's just not wanting to work.
That's strange. Okay. Well, let me take it from here. Okay. So where was I? Tumor lysates. So I mentioned that those clinical trials largely failed for reasons that there was antigenic competition, and also what you see in the tumor lysates that you have a lot of immunosuppressive signals from the tumor that kind of dampen the immune system. Now, about 10 years later, there was a seminal discovery when people discovered the first cancer test engine that was also previously presented by some of the speakers before, MAGE-1. Followed by a number of other cancer-tested antigens. Now that initiated a whole surge of cancer vaccine companies and hundreds of clinical trials being performed with various forms of these cancer vaccines. Unfortunately, all of those failed again for reasons that they kind of lacked sufficient immunogenicity. So the problem with these antigens is that they're expressed to some extent in the adult organism. And so you get some degree of self-tolerance. The immune system is recognizing the self-antigen, so it's kind of getting rid of those immune cells that are forming an immune response. Another reason was that these cancer vaccine products also contained very short peptides, so between 9 to 11 peptides. And actually, Kees Malief, a scientist, but also the other scientists in the field, he found that if you use short peptides, that these peptides are very insufficient. They're actually not presented in MHC class 2 molecules. Now, MHC class 2 molecules are important to get the CD4 T helper response. And with that, you get more, you get actually better CD8 T cell response and also memory formation. And you also kind of involve the innate immune system because it really turns out that you need a full army of immune cells to combat the cancer. So based on that, we started a company together with Kees Malief called ISO Pharmaceuticals in 2004, which we also managed in the initial years ourselves, where we worked on a synthetic long peptide vaccine, so longer peptides that are actually very efficiently processed in both MHC class 1 and 2. So you get the CD8 and CD4 T cell response, where the company focused on a cancer vaccine targeting human papilloma virus, E6 and E7. And these viruses are known to cause cervical cancer and head and neck cancer, amongst others. Now, the company actually reported the first clinical results of their vaccine in the New England Journal of Medicine in 2009, where they showed that, quite excitingly, the product was able to more or less cure patients suffering from pre-malignant vulvar cancer in about half of the patients. So really durable remissions for more than 2 years, which was associated with also a strong level of T cell activation and proliferation. And now a year later, Provenge came on the market, so the genetic cell vaccine that was covered before. And then we saw the advent of some of the checkpoint blockers, like CTLA-4, PD-1, anti-PD-1. And so that kind of all transformed the whole oncology treatment landscape for many tumors. Actually, a year later, there was a seminal discovery that when scientists started to look into tumors of patients that either responded or not responded to PD-1, and they found actually that those tumors that had a very high tumor mutational load, such as, for example, melanoma due to UV light causing a lot of mutations there in these cells, that actually those tumors quite often respond pretty well to anti-PD-1. And the reason for that is that if you look in those tumors, there are a lot of T cells in there reacting against the new antigens that are formed by these mutations. Now that kind of fired off a whole new excitement in the field, and companies like Nuon Therapeutics were formed. BioNTech also played a role there in testing these personalized neoantigen vaccines in the clinic. Unfortunately, also these vaccines were unsuccessful. I mean, they were safe. That was the nice thing. In general, cancer vaccines have been found to be pretty safe, but unfortunately, they did not induce sufficient immune responses in patients, and not leading to durable remissions. So whether or not we'll are on the slope or on the valley, we don't know. We do see still a lot of coverage still in the news from companies developing new antigens. So who knows? Let me see if I can move to the next slide now. Yeah.
I'm sorry to interrupt you again. I think it zoomed in quite a bit into your screen, so we can't see all the information. And I mean, we have a few people in chat who are prepared to hack your computer. We don't want that. So I think someone has said that should be a lot better. They've said in your top right corner, there are a couple of icons. There's a box with an arrow pointing up. That has been suggested. Otherwise, I think if people are happy to just to see it as this, so we've got all the information on screen, that should be fine.
Yeah. Let me see.
Or it might be towards the left, sorry, on the left. On the left, so on the red bar that you've got the top.
Yeah.
Okay, try that one. Don't worry, I think there might just be a glitch with PowerPoint and Zoom just not liking each other at the moment. So this is fine at the moment, as long as we can see. You might want to zoom out just a little bit more, just to allow us to be able to see the full scope of the screen, please, if you would.
Yeah.
Perfect, thank you. Thank you.
What about that? Is that better?
That should be fine. I mean, you're welcome to zoom back out a little, but that should be fine. I think we can see. Perfect, thank you very much.
Okay.
Apologies for interrupting, please
continue.
No worries.
So what are the causes of the cancer vaccine failures? Well, there are basically three reasons. One is the wrong choice of the antigen, wrong choice of the platform, and as well as the clinical development strategy, which is not appropriate. So I will be discussing these topics in more detail in the following slides. Now, first, just to kind of review again, what is a cancer vaccine product actually? Well, it's a rather complicated product consisting of 4 components. One hand, you have the tumor antigen, but you also have the way to provide antigen, an RNA or DNA or a bacteria that can be provided by a virus or bacteria. And you also need a powerful adjuvant, like a toll receptor agonist, for example. Now, in my talk, we'll be focusing only on the tumor antigens and the formulations. So these are the 5 main categories of tumor antigens that are being used in the vaccines of today. And these are some of the main criteria, considerations that these antigens carry. Now, with regards to the overexpressed antigens, such as HER2, for example, or the differentiation antigens, such as GP100 and melanocytes, you also have the problem of a very high degree of central tolerance, which you need to overcome by bringing a very powerful adjuvant. Also, if you're actually able to break tolerance there, you risk autoimmunity. And we actually know that from trials with CARs directed against these same antigens. Some clinical trials have been done there, and really patients have died because of autoimmunity against those targets. The cancer-tested antigens look a bit better from that perspective. They're more specific of tumors. I mean, you still have some expression in the testis, but I guess you can live without it at some point in your life. And there's a more variable degree of central tolerance to those. With regards to the tumor-specific antigens, well, oncoviral antigens, like human papillomavirus, is a very attractive target, I think, because it's clearly foreign and is expressed in all the tumor cells. HPV is causing that cancer. About 10 to 20% of cancers are caused by viruses, so there's a substantial patient population that you can address there. And you can actually give the vaccine off the shelf. The shared neoantigens are antigens like frameshift mutations, re-expressed retrovirus, what you see a lot in some tumors, but also like driver mutations in cancers are shared between patients. They don't occur a lot, but there's a sort of a minority there. And of course, the private neoantigens that were also covered today. So these are truly personalized therapies where you look in the individual tumors of patients and you predict the immunogenic epitopes, make a product, and then give it back to patients. Of course, it's a product that cannot be provided off the shelf, so you really have to make it tailor-made for the patient. Now, what are the most popular antigens these days? So this is a cartoon which depicts the top 15 immuno-oncology targets in 2019. The yellow bars are the cancer vaccine trials. Overall, we had around 800 cancer vaccine trials in 2019, so 30% up compared to 2017, actually. So it still seems the field is moving up. And the most popular antigens there are HPV, so human papillomavirus, followed by HER2 and some of the other cancer associated antigens. On the right-hand side, you can see the personalized cancer vaccine trials, and you can really see there this is really a minority of all the cancer vaccine trials ongoing. So what are now the ideal specifications of the antigen according to in our view? Well, if you have the luxury to actually choose, you want to have something that is truly tumor selective, that is strongly and homogeneously expressed, and also clonally inherited, which meaning that all the tumor cells carry that antigen, because otherwise what you will see is that the cancer cells that don't carry the antigen will actually just escape. Another way to overcome that is actually by targeting multiple antigens in the product, so you get coverage in all the clones. So that's another approach. You really have to consider putting epitopes that confer both MHC class 1 and 2 loading, so you get the CD4 and the CD8 T cell response, and then therefore you really get the memory response there as well. The more the antigens are shared between patients, then of course you can start to think about giving off-the-shelf therapies, which of course have a much lower cost of goods. Now, these are some considerations with regards to the platform selection. I will be focusing on the nucleic acid vaccines and the peptides here. So this is roughly the pros and cons of the approaches. Clearly, there are a lot of biotechs there that are trying to overcome some of these limitations, but it gives you a rough idea of the pros and cons there. Now, with regards to nucleic acid vaccines, you can use naked DNA and mRNA. And the nice thing about that is it actually has intrinsic adjuvant activity because it triggers the toll-like receptor agonists on the APCs, so the antigen-presenting cells. Unfortunately, it's actually very hard to get sufficient amount of DNA and RNA in the APC. Cells don't really like to take that up, and if they do, the DNA actually has to migrate all the way to the nucleus to get transcribed and then move out again to the cytosol and then be translated in the protein. So there are a lot of steps there. So what we also see that this typically delivers much reduced immunogenic responses. There are some certain safety constraints. So DNA, you always have the risk of viral genome integration. Cost of goods, however, are very low. DNA is, of course, very cheap. Our mRNA is slightly more expensive. A way to overcome these limitations is by encapsulating mRNA and DNA in nanoparticles. You kind of lose some of the intrinsic adjuvant activity there because you don't trigger the toll-like receptors on the plasma membrane. However, you can still engage with those toll-like receptors on the endosomal membranes. But clearly, you need a technology that allows you to actually release these antigens from the nanoparticle. The good thing about nanoparticles is that they have a natural distribution to APCs, so they're readily engulfed by DCs and macrophages. So you get good coverage there.
Dr. Goetjes, apologies for the interruption again. May I ask which slide you're on at the moment? Because we can see slide 5.
Slide 5. I'm at slide 11, so.
Right, don't worry. How about, let's take a minute. Would you like to send the presentation to us? We'll see if we can. So let's just have a 2-minute break. And if you could email, so if you want to stop presenting, if you could kindly send the presentation over to us, and then we'll try and get that running on our end. So if we can just have like a quick 2-minute breather, we will be with you again. I think there's quite a bit of interest in your presentation, and of course, we would definitely want to see the supplementary information that comes with it on the slides. So if you could kindly just send that to us, we will have that ready for you. So everyone, if you could just bear with us just for one moment. If you would actually, someone has suggested if you wanted to save it as a PDF for yourself, that could also work. And then you can go through each page, whichever is easier. So no pressure, we'll give you a minute.
Yeah, I'll send to Andre, it's probably easiest. Let's see. Andre, it's on your way.
Yep, I can hear.
Andre, are you happy to drive the deck for us? That's a really interesting suggestion that we should save it as a PDF. Apologies if I'm not pronouncing your name, Vladislav. Do you want a job as an IT technician? I don't know. Send us your CV. I'm sure you're way overqualified for it, though.
Well, I have it as a PDF now as a backup as well. So
if you want to try it as a PDF, that may work as well.
Okay, yeah, let's give it a shot. Let me see. Where do we have it? Yeah. Is that better? Then I have to get it to full screen.
I think there is a way. Try F5 again, or Shift F5. That usually will allow you to do it. I think there is a full screen option there somewhere. If anybody wants to suggest where it would be, try Control L. try pressing Control and then L. Wonderful. Thank you. Yeah. Please continue.
All right. Sorry about that. Oh, let me okay, pick up the talk from here. Okay, so where was I? Well, I was discussing the nanoparticle encapsulation of mRNA and DNA. So clearly, the advantage of a nanoparticle is that you actually get very nice distribution to the APC, so you get good coverage there. Limitations are that you get a little bit less efficient MHC class presentation. Cost of goods are, of course, slightly higher than naked DNA and RNA. Another way what you can do is actually put the antigen in a viral vector. Actually, viruses have a very intrinsic adjuvant activity. The problem is that you will get a presentation of antigens from the viral vector, but also from the antigen. So there's a lot of antigen competition, actually the antigen that you want there are kind of underrepresented in the MHC class molecules. So that's some of the limitations. There's safety constraint there as well. I mean, you can get viral integration of the product in the genome, and the cost of goods can be actually quite substantial. Now, peptides, the short peptides I discussed, so they don't get efficiently processed in MHC class 2. And whereas that the synthetic long peptides are actually processed both in MHC class 1 and 2. Now, peptides don't have intrinsic adjuvant activity, so you need to come with an adjuvant. They have a good safety profile though, but the cost of goods can be pretty high, especially if you're going for the truly personalized vaccines, then the costs are actually quite high. Now, what to look for in a platform? Well, if you're developing in a cancer vaccine product, what you want to see is a very strong single agent activity of your cancer vaccine in the tumor model of your target indication, alone, possibly in combination with anti-PD-1 in established tumors, or really polypal tumors, because it's much easier to cure cancer in the preventive setting. So you want to do that in the therapeutic setting. And you want to see that remission durable, so at least more than 100 days. That also the anti-tumor response is CD4 and CD8 T cell dependent. You want to see both CD4 and CD8 T cell responses, hallmarked by type 1 cytokines, some reminiscence of sort of an antiviral response. And you want to see memory T cell formation also long after you've done the last injection. Pharmacodynamic data for the full circle of cancer immunity cycle, meaning APC activation, T cell priming, T cell activation, and then T cell migration to tumor microenvironment, killing of the cancer cells. What's very important also is to get the timing right on and sequencing in the combination therapy. So when will you be providing the vaccine and when the boosters? When will you be adding, for example, checkpoint in there? That can have quite dramatic consequences if you get the timing wrong. For example, if you in some cases they found that if you administer PD-1 before the cancer vaccine, that you get actually kind of make your T cells exhausted. So that's something that you obviously don't want. Nice to have there is to show epitope spreading, which means that you get T cells reacting to antigens that are not in the cancer vaccine product. And also showing some human ex vivo data, so APC activation in lymph nodes, for example, from patients. Now here we'll be sharing some considerations with regards to clinical strategy. Now clinical trials have shown that the cancer vaccine product on its own will likely not be successful in almost all cancers. I think isopharmaceuticals product is only one example that I know of where the cancer vaccine product on its own was efficacious in this case, a pre-malignant disease, so not a fully malignant disease. So in all other cases, you need to start to think about combination therapies. And what makes a lot of sense, of course, is to combine the cancer vaccine with PD-1, because PD-1 acts on the T cell, and the vaccine acts, of course, on the APC. So that should be a match made in heaven. Now, if you have a tumor that has a low number of new antigens, you can also start to think about using chemotherapy to liberate more new antigens from the tumor, or for example, using radiotherapy. You could also think about enhancing the access of lymphocytes to the tumor microenvironment, for example, by using angiogenesis inhibitors. Now, what has become clear over time that not only the tumor is very heterogeneous, also the immune cells in the tumor microenvironment are also very heterogeneous. So this is a paper that came out about 3 years ago, where they looked at 10,000 tumors and where they classified the tumors according in 6 different profiles. So the C2 and the C3 tumor categories, so interferon gamma dominant and inflammatory categories, are the so-called hot tumors. And these are typically the tumors that actually respond, well, are most likely to respond to PD-1. Whereas the C4 and C5 tumors are the so-called immune excluded and immune desert tumors. And these tumors are generally don't respond to PD-1 because there's simply no lymphocytes in there or an existing TIL therapy reacting to a neoantigen. Now, if you look at the individual tumors on the right-hand side, you can see there's a lot of heterogeneity there. For example, if you will be going for bladder cancer, which is the second one from the left, well, you can see that cancer actually has 4 different categories. So if you're going for this tumor type, you may not want to be going for all of bladder cancer, but you may want to be going, for example, for the hot ones there. You could be going for the other tumors that are more cold, like GBM, for example, but then you need to start to think about a second therapy that deals with that. And one thing you can do is to combine it, for example, with chemotherapy. This is a trial that Pharma did in cervical cancer, where they treated patients with carboplatin and paclitaxel. And what you can very nicely see that if you treat those patients, you get a very nice reduction of the MDSC compartment, which is a very immunosuppressive cell type, which generally increases the immunogenicity towards HPV peptides, but also to microbial antigens. So it kind of relieves the immunosuppression there. Some other things that we learned, well, it's becoming apparent that if you are that you have a patient that is very advanced in cancer, meaning a lot of metastases, large tumors, that those patients are less likely to respond to cancer vaccines. I mean, there's just a lot of heterogeneity and a lot of immunosuppression going around in this patient. Actually, some data here from BioNTech on the right-hand side, where they found a correlation or trend to correlation between patients that had a less bigger tumor and a better response. This is actually not a complete surprise, because it's already known that if you apply checkpoint inhibitors in the adjuvant setting, so right after surgery or chemotherapy debulking therapy, that actually works a lot better. So what are some of the recommendations that we have for clinical PK and PD? Well, first, you of course also have to think about how you want to administer the drug. So intravenously, intradermally, subcutaneously. I think what's important there is that you get very good coverage of your dendritic cells, which are the most important APCs, particularly the DC1 cells. You want to see that the product actually is clear to the lymph nodes where the priming of the T cells takes place. And you want to see that these antigens are actually displayed on the surface of the APCs. And whereafter they should prime the T cells. So you want to see activation of T cells, good Th1 responses, and you want to see strong avidity, meaning that a low amount of peptide already induces a very strong immune response. Then if you get the clinical results, you want to be correlating the immunogenic responses to clinical responses. So on the left-hand side is an example of a study that ISA did, again, a cervical cancer, where they looked at strong responders and weak responders, where they found a very significant enrichment of CD8 and CD4 T cells in the strong responders. Whereas in the weak responders, they found immunosuppressive Tregs. Now to conclude my talk, what are now the biomarkers that will enhance the probability of your success? So firstly, going for an antigen which is strongly clonally expressed. Selecting more than one is probably a good idea. going for those tumors that have an inflamed signature, although this is also an environment which is very competitive these days. Those tumors are also much hallmarked by immune cell infiltrates. You might consider going for tumors that are absent for immunosuppressive cells, or you have to think about a strategy that actually deals with that. Be also mindful of excluding those patients that have resistance-driving mutations. So cancers, for example, can reduce expression of MHC class molecules and thereby prevent presentation of their new antigens. But you can find those patients by sequencing them and filtering them out. Responsiveness to anti-PD-1. Clearly, so that's also a very competitive market. A lot of people are trying that. So if you are going for patients that are anti-PD-1 resistant, well, it's probably advisable to go for those that have an inflamed signature in the tumor microenvironment. Immune fitness of the patient, that also helps to enhance the immune response. And like I just mentioned, low tumor burden also enhances the chances of success. So with that, I will end my talk, and I wish all the entrepreneurs that are listening a lot of success.
Thank you very much, Dr. Goetjes, and thank you very much for accommodating the technical issues and for updating the presentation for us as well. So yes, we do have several questions already forming there, which will take part in the Q&A in just a short while. So please do keep the questions coming, keep your comments coming as well. We would really like to invite as much conversation as possible. And finally, I'd like to introduce the next speaker, which, bear with me just one second. Yes, he will be our last speaker for today. Christopher M. Coley, Marketing Development Manager of Digital Pathology at Panasonic Corporation. He will be joining us with a talk from 1 week to 1 day. Take control of your research by automating tissue microarrays, multiplexing, whole slide scanning, and AI analysis for enhanced bioinformatics. Christopher, over to you. We'll just give you a moment to get your slides ready as well. We can't see you as yet, but we'll give you a moment.
I'm afraid I'm having technical difficulty. You're going to have to give me a moment. I apologize.
No, that's fine. If you prefer, if you've got some slides or visuals, you can send them over to us and we can load those up for you if you would prefer. Or if you need a minute, we can, if you would like to leave and rejoin, if that helps.
Yeah, let me see if I can pull this up and I'll call back in. I apologize.
No problem. No problem. Speak with you soon. Thank you. Right, we'll just give Christopher a moment to rejoin. I can see some really interesting questions here. So please do keep them coming, and your comments as well in chat are very supportive. Please do comment on any of the material that you've seen or any of the comments that have been made as well from the speakers. I'm sure they will be very much welcomed, and we're very interested to hear what you think and your own experiences. I could see from chat there were a couple of comments from experiences. Someone mentioned earlier about their daughter who I believe in December will be receiving such vaccines. So these are really, really interesting. It helps us give a real life perspective and helps us to really empathize with the material. Oh, Christopher, I can see that you're back. We'll just give you a moment.
Yeah, I'm not able to pull them up, I'm afraid.
If you would like, if you can send that over to us, we can pull that up for you if it's easier.
Yeah, let me give that a try.
Yeah, no problem. That's absolutely fine. We'll just take your time. We will be here. What I will do in the meantime is just give everybody a quick refresh of our panel. And where they're from, their roles. I think we've had a really interesting spectrum of topics here as well, particularly from the perspective of technology, to look at what's coming up next, the future, the next few years. And also it's quite interesting to have seen the landscape, the visualization as well. I think that was quite powerful. And I do believe that will be available to attendees as well. So the link should be within chat. I think it is www. Let me get that for you. Let me get the full link. So, yep, www.deep-pharma.tech. And again, please do review all the links that we've sent to everybody in chat as well. So let's see, Christopher, did you send the presentation, was it to Andre or to Helen? I think we've lost him, bear with me.
All right, I've tried to send that over via email.
Sure, who did you send it to?
To Helen's email.
Okay, Helen, if you could kindly load that, otherwise you can send it to Katrina, who has kindly volunteered to do that. Christopher, are you able to see the screen at the moment? Christopher, are you able to see the screen?
I can see the screen now.
Lovely. Okay, so if you bear with us, what we will do is I will stop sharing and we will have that loaded up for you. And when you would like to move on to the next slide, just please do say next slide, please. Okay, so just a moment, please. We'll see if that's being loaded up for you. Okay, so it's currently being uploaded right now. Please do keep your comments coming as well. It's really interesting to see where everybody is. Let us know which corner of the world you're joining us from. I myself am from the north of England. I'm not from London, as everybody would normally assume. I am from a little area, a little county called Yorkshire, which is in the north of England. I usually do have a very heavy Yorkshire accent, which I'm hiding for several of you, as it will be easier to understand without it. Where are our speakers from today? That would be interesting to know as well. Dr. Goetjes, where are you at the moment?
Still on.
Ah, whereabouts in the world are you right now?
Amsterdam.
Amsterdam, ah, lovely.
Yep. Yeah, it's not too bad here.
Sorry?
It's not too bad over here. Well,
I think we share, I think where I am, it's almost level with Amsterdam. So I'm in the north of the UK, well, not all the way up north. So we probably share the same weather.
Yeah, yeah, it's
sure.
Amsterdam weather kind of phenocopies UK weather.
Yeah. How about the rest of you? Lovely. I think we are ready. We will certainly come back with our little geography Q&A. So we are ready with the slides. Yes, Christopher, please do just advise when to skip the slides. Thank you very much, and looking forward to hearing a little bit more. I will just, I think I've already introduced you, but I'll just do that again. We've got Christopher M. Coley, who is the Marketing Development Manager of Digital Pathology at Panasonic Corporation, and his talk, From 1 Week to 1 Day, Take Control of Your Research by Automating Tissue Microarrays, Multiplexing, Whole Slide Scanning, and AI Analysis for Enhanced Bioinformatics. Over to you, Christopher.
Yeah, sorry, I'm still having the difficulty. There may be audio attached to these slides. Can you try to play from there?
Sure.
Thank you.
Okay, So Helen, I don't know if you caught that, but there will be audio that will be attached to the slides. And you'd like us to play the audio, yes?
If you can, I'm not able to access it from here.
Okay. So we're on the first slide there. So Helen, if you're able to play the slide, the audio.
So to be honest, I do not know how to play the audio.
No problem.
If Christopher,
if you're happy for us just to proceed with visuals, and then we can always try and incorporate the material with all the materials that we'll share later on as well. I believe there will be a video as well, so we will try to incorporate that for you.
Sure, go ahead. I'll see what I can do from on this side. I apologize.
Not a problem. Thank you very much. Off we go.
Yep. So basically what we're looking here is a lot of these processes that you can see in the laboratory when you're looking to do some of these research, so they're very manual. And it starts with making tissue block microarrays. And in order to do a comparison, what we've done is we did 100 tissue cores, and we did them in an automated, and we compared that to a manual process. And we were able to program the instrumentation to do this in an automatic way, set up the distancing, where the cores go, keep up with the data that goes along with those cores, the position from the donor blocks that as they come across, and be able to later go back and pull that information. It also puts them in a very precise depth level, depending upon how many sections have been taken from the donor material blocks, so that all of the cores are positioned in the same way. And if you do this in a manual method, it's very tricky, and you have to have a good skill set in order to be able to do this. It's extremely time consuming, so automating this is helpful. We've also taken that automated into doing an automated multiplex staining for immunofluorescence, instead of having to do a manual method, then scanning those images, those slide images, and then taking the end of those results and doing algorithms with our AI partner to count the cells and be able to do a precise data return. So next slide, please. And as you can see here, the equipment from left to right is the 3D Histex Tissue Microarray Master 2, which is a small, great automated device that we'll showcase. In the middle is the LunaFluor LabSat. And then on the right-hand side, we're using the 3D Histech MIDI2 12-slide fluorescent and bright field scanner. This one in particular uses a Lumicore spectral light source. Next slide. So automated wise, starting from left to right, we're able to achieve more than 450 cores in a single block. These are used using enhanced punches and drills from 0.6 millimeter, 1 millimeter, 1.5, and 2 millimeter choices. And again, you can establish where on the block you want these cores to go. And you have the option to go in and do very precise coring, as you'll see in upcoming slides, to select the precise cells that you want cored out of the paraffin blocks. Next slide. So as it says, the TMA Master 2 does have a small footprint for valuable lab space. It's an easy customizable layout in the software that you can see with the diameter shown, and you are able to, over the paraffin block itself, overlay the image of a scanned slide that matches, whether it's immunofluorescence or it's chromogenic IHC or the H&E, and designate very precisely the groups of cells of viability that you want. So if there's areas that have perhaps necrosis, or they have fibrosis or other features that you don't want in your sample set for the cores, you can choose the most precise viable cells with the best staining for it. It does read the barcodes and the faces and take images of those that go into the spreadsheet and the datas to be culminated later, so that you can go back, and as you go through the cores and break them down for the sample staining, it will show you which blocks it came from and which positions in the tissue it came from. The next slide. So you can see here in the slide layout, I can completely compose my grid. It doesn't have to be as complex. This is an example. We do have several customers with up to 500 cores in a block. You can choose the layouts left to right. You can choose the distance between the cores. You can choose the depth of insertion into the block. All of this is an automated approach that saves a tremendous amount of time over doing the manual process. Next slide. Here you can see the slide image overlay, which is a really unique feature as opposed to some of the other systems which can be semi-automated. This is phenomenal. Instead of having to try to eyeball, if you have a block that is appears homogeneous and you cannot tell the difference of one tissue area from the next, let's say it's just a block of liver, for example, you can take samples from anywhere in there. But if you have a block where there's multiple tissue types in the paraffin embedded sample, this allows you to overlay the H&E or the IHC, select those particular cores, and the circle will match the size of the cores that are going to be taken. Select very precise region of interest and insert those in the area on the recipient block that you want. You're also able to take those cores and put them into a PCR tube for molecular testing down the road if you don't want to put them into a block. Next slide, please. Again, this is good data tracing to go into the reports. It does capture the face. If your donor material blocks don't have a barcode or a printed material and it's just a handwritten, it still captures that. And if it scans that block again, no matter what, if it's barcoded, printed, or handwritten, it'll go back through and say, I think I've scanned this block before, show you the previous scan and be able to allow you to say, yes, I've taken cores from this block before. So from a data saving export standpoint, it's phenomenal in being able to keep up with which cores taken from the donor blocks go into which recipient block positions. Next slide. This is part of a family, right? So the TMA Master 2 holds one recipient block and up to 4 donor blocks. If you're looking to just make more generic type samples, you can change that configuration. You have 5 positions to do with as you please. It does a phenomenal job of drilling into the holes. You can set this up and count down, leave it for overnight if you have very complex. If the 5-block system is not good for workflow and you're making something much more complex like some of our pharmaceutical companies use, the item on the left is our TMA Grandmaster, and it's a real workhorse for people that are doing 400 plus cores in a particular recipient block and want to make lots of recipient blocks. It'll hold 72 blocks total. It uses a drum that rotates for that. It'll have 12 recipient block positions, and it'll have room for 60 donor block materials. So very complex if you want to do very specific arrays and tumor array panels, and that saves you from having to load and change out the blocks in the other workflow. The TMA Master 2 is really good for a lot of our clinical customers that maybe want to make disease panel state blocks for maybe a breast cancer or something along those lines, or for special stains where you really only need a handful of blocks for donor material and nothing as quite complex as you would see with the TMA Grandmaster. Okay, next slide. The TMA transitions over to the multiplex staining. So once you have that multiplex block array ready to go, you can deparaffinize, go onto the machine, which is the Lunaphore LabSat, as you can go ahead and proceed to the next slide. So this is a phenomenal way of time saving. These multiplex traditionally can take days to produce and require an awful lot of time of checking, rinsing, going back and forth and through here. And this simply allows you to load one slide at a time and do the multiplex staining and allow the end user in the laboratory to move on to other tasks while this is automated. Next slide, please. So this is designed for the chromogenic formalin-fixed paraffin embedded IHC, frozen section IHC, and then the fluorescent stains from multiplex traditional IF, frozen section IF. So it's a real workhorse for these types of tools, whether you're doing a quick check on something off of a cryostat, such as a muscle biopsy or renal biopsy in a transplantation case, or if you want to go on to doing a multiplex off of a formalin-fixed paraffin embedded. So these have lots of applications to be used. It's very rapid because it uses microfluidics and pressure in order to increase the time savings that you would see off of a manual type system. It's extremely precise in the fluid reagent management and uses a small amount of reagents when you're going through the system. And you can tweak the customized protocols for optimized staining for various multiplex kits that are on the market today. The next slide. So again, the microfluidics and rapid is the real key here that you're looking for. These biomarkers in development allow you to have precision control. So if you're developing an assay, you're able to do this quite quickly. You're able to save precious reagents or antibodies if you're developing your own products, and allows you to have that highly reliable reproducibility and control to bring these types of stains into your lab. It is a skill to do these types of stains manually. It takes a fair amount of training and time, and the reagents are quite expensive. So this is a really good way of making the most out of the reagents that you have to order for these types of tests. Next slide. So the ultra rapid turnaround time is why it's so fast. So again, this goes over to the flash fluidic exchange. There's the slide that sits into the apparatus. There is a pressure control delivery of the reagents that allows them to go through and gets the interaction with the epitopes and the markers on the slides very well. And then you can control those incubation steps very precisely, as you can see here, to temperatures above 100 degrees without affecting drying or evaporation or inconsistency across the tissue on the slide. This keeps everything at a nice homeostatic environment so that you're able to conduct these things reliably over and over again. Next slide. And the results speak for themselves. We've got phenomenal results with various multiplex and even single staining on here, so that you can see when you go into scanning later on, you're able to go through and get good, consistent, quantifiable results. So the staining intensities will be the same over and over and again, so it doesn't throw off or require additional calibration steps when you're staining to be able to quantify later on, which adds a lot more time down the road. You're able to get all of that with the tissue that fits up underneath the chip. So you've got rapid incubation and a lot of tissue surface area that you can control for that. The next slide. So the fast fluidic exchange technology, you're able to do this with multiples. You can do this with the TSA kits. You're able to do this with doing sequentially and specific antibody cocktails. So if you're doing biomarker discovery, you're allowed to do a lot of these markers on here with the TMA slides to get a wide burst of array sample stainings or early on, and be able to taper that down as you whittle away for the best staining sets and the best reactivity for the tissue that you're going to have. So you're conserving tissue, you're increasing your availability of sample set, and you're decreasing the amount of time that it takes to conduct that TMA block and the reduced amount of time to perform these very lengthy staining protocols. And next slide. So here's an example. This is formalin-fixed paraffin embedded tissue. This is a human tonsil section, and we used a six-plex marker with DAPI with FOX, PD1, CD8, PDL1, CD68, and cytokeratin. And this was a 4-hour, 37-minute protocol that we ran, TSA-based chemistry with phenomenal staining results across the entire sectional cores. And again, we did this with 100 cores for this particular application, so we could do a side-by-side comparison for our data coming out on the other side here. And the results are extremely good. We're able to get these nice patterns in here because of how the arrays are laid out, and then later go back in and do the quantification steps. Next slide, please. So we move on to the multiplex to the whole slide imaging on the MIDI-2 that you can see on the right-hand side with the fluorescence application. Next slide. With the scanning on here, you can see the bar at the bottom to the left and the right. You're able to put flat trays for these aqueous mount slides for immunofluorescence. And there are shields to protect from the light that's in the room so that you're not sacrificing the fluorophores as you're doing the imaging. Next slide. So this is a great concept. These instruments in this MIDI-2 come in multiple configurations. They're a small footprint for a good laboratory space. You're allowed to select 1 or 2 objectives on these instruments, actually 3 different objectives now, a 10x, 20x, 40x, and with camera adapters, you're able to expound those up to 96x native resolution before you do any digital enhancements based on the cameras and the lenses on the optical pathway down into the sample here. In the light engines that you're on here are specifically designed to be able to pick up based on the different immunofluorescent multiplex panels that are on the market. And there's plenty of space on the turret for 9 filter cubes. And each of those filter cubes are able to hold anywhere between 1 and 4, so single to quad band filters. So you really have a full spectrum of filters to your availability at any given time without having to go in and make manual changes to the instrumentation. You're able to also, once you do your scanning, as it sees here, you can have several images pulled up at one time. You don't have to review one at a time. You're able to pull up the different channels individually, and then as you want to increase them into a single view of it. The slide viewer pairs well with image analysis platforms. We work extremely well with a lot of the established image analysis and those that are our own creating and our partners. Next slide, please. So as you see here, it's a very easy click-through menu. You can select the channels that you want up front, pick the filters that you'd like to have in here, and then you can do an automatic or manual exposure. And using the dark field preview, you don't have to go in and focus on all of them. It will go through the spectrum using DAPI, and it will make a correction so that you're not getting overexpression in some and underexpression in others. It tries to make all of the excitations even so that when you view the image, everything is on the same level of intensity and you're not muting out anything and you're not being blinded by autofluorescence or any of that sort of thing. It calibrates those things so you don't have to go back and do it. As you're developing protocols for an antibody cocktail, you're able to go in and change those manual exposure times and then save them if you wish as a protocol that you can repeat without having to make additional manual adjustments. The next slide. So once you've done this with the TMA module, it takes the grid of cores, and instead of having to view them all as one slide, it will take each of the cores and make them a separate image for review. So you're able to take the 400 plus cores that you have in your array. It will take each one as a separate entity for you to evaluate and do the analytics on, so that you're not having to do them on all of the cores at once. It allows you to break them down and keep that data tissue by tissue, core by core from the donor blocks, so that you can see exactly what the tissue types were and how those resulted in your staining. The segmentation samples based on the analysis and measurements are really key here. So you're able to go in and really count and start doing the deep dive into it. And if you need to do an automated rescan because you're changing some parameters on those results, you're able to go in and easily take each of those individual cores as opposed to the entire block and rescan just those sections. Thanks. Next section. So as we go through this, Quant Center is a software that 3D-Histic has various features that allow you to do image analysis as you define. You can do based on specific cell types. You can do the entire slide. You can circle specific areas that you want to do and look for very defined specific features as you want to look at morphological changes, staining pattern changes, quantification and scoring of specific cells. So you're able to set each of those up and then run them on here, both bright field and immunofluorescence. Next slide. So the HistoQuant is one of those algorithms that we use, and it allows you to do segmentation and measurement based on the colors of the cell components. So in the fluorescent area, it's very easy to do because I've artificially assigned those various colors of blue, green, yellow, and red on the filters, select the area that I want to do, and go ahead and churn that out, quantified based on the percentage of the sample that is being evaluated, or the whole slide, and the specific channels and colors as we see fit. And we're also able to negate the ones that we're not really interested in, that maybe DAPI is just providing a morphological background. You don't have to include that in the sample counts if you don't want to. So it's a very nice customizable tool. And again, these can be saved as protocols. Next slide, please. So from whole slide scanning to bioinformatics, we're partnered with Iforia, which has a great way and unique tools to do artificial intelligence above the image analysis that we described in the Quant Center. Next slide, please. So on the research end, we've got the Iforia Hub. This is all a web-based tool. We use Iforia Create, and you're able to go in and use the staff or your own, after training, go in and take your sample sets from easy to complex and have these protocols developed as you partner with the company to be able to perform that AI supported analysis. It just is extremely customizable based on the layers and the types of cells you're wanting to look at in both bright field and fluorescence. Next slide, please. So as you can see here, this is an example where you've got, you can annotate and train the AI based on the level, the identify, quantify, or measure the features of interest. You can pick as many layers as you want to here. You can see multiples. You can have a stroma that's particularly labeled in green, and you want to be able to see what the percentages of that. And then maybe I have an area of interest, a particular tumor type or disease state. And within that, I'm able to also check out different variations of those cells, very specific patterns of those cells, depending upon what disease state I'm looking for, whether it's going to be cribiform or sheets or various different patterns in those tumor cells. And as I add those layers, it adds to the quantification output that I'm able to do. So you can start simple and build on the complexity as your research grows in that particular set of slides. Next slide, please. So here's a more complex based one. This is a cloud-based, of course, deep learning algorithm that's being used. You can define the image analysis that you need, create your bespoke AI model, analyze the images in the model. It's extremely easy to use. In this particular instance of a bright field, I'm able to select in here from the brain tissue the various cell types that I want to do, and then furthermore go back in and sift for specific abnormalities or styles of those cell types, and very clearly see while I'm setting up the layers how this is going to look as I go through this. And it's extremely rapid technology. There's no need for any coding or prior knowledge. You basically walk through and select visually the areas that you're wanting to do, and then you're ready to start training on your models and kick it out. Next slide. So here's a good example here for the unlimited channels. Again, this is a tool that is as complex or as simple as you want as you're going through here. I can select the entire section on the slide. I can go into very specific regions of interest under high magnifications and get the details as specific as possible based on what I'm looking for in my analysis. And that's the nicest thing, is this isn't a tool that's preset. This is a tool that's important to what you're looking for in each slide, in each set of research. Next slide. So with the this particular one, we did the 6 fluorescent channels for the multiplex. And as you can see here, we're able to simply go in and define what each of those are, what types of cells we're looking for. You can see the annotation, the percentages, and there's a whole host of tools and snapshots that I can make in different regions as I want to look at these in different layers. I don't have to look at each of the stains at the same time. I'm able to filter them out, choose it, get a really crisp view of the one channel that I'm looking for and highlight those cells that I really am interested in doing, and then build upon the next layer. Next slide, please. So again, this is a huge impact for the research. The automation of these manual processes on a limited amount of footprint space in the laboratory is really important. It's a huge control. You don't, laboratory researchers that may have not been able to do this in the past. Are you able to hear me?
It did cut out, but we can hear you. Hello?
Can you hear me now?
Yes, we can hear you.
Sorry about that.
That's
So
okay.
basically it's designed to do an awful lot of control so that you can bring these tests into your laboratory if you've been sending them out prior to that. And it's able to save an awful lot of time and give you an awful lot of consistency with the end goal of being able to consistently get your data quicker with less time and reagents. I'm not able to see the screen anymore.
So we're currently on the Impact Your Research slide.
I can see it now. Thank you. All right, you can go to the next slide. All right, I think that might be the.
Yes, that's the end, I think, of the slides there.
All right, apologize. Thank you for your patience while I was going through these without having the script in front of me for whatever reason, and look forward to whatever questions come up.
Not a problem. Thank you very much, Christopher. It was really insightful. And again, thank you for just being good and just optimizing and just allowing us to adapt and drive that deck for you. So yes, thank you so much to all of our speakers. Much appreciated for all your contributions. So now what I would like to do is welcome our speakers. If they're able to, please do turn on your microphones. And if you would like to and are able to, please do turn on your cameras as well. And now we will be having a roundtable discussion. Lovely. Okay, if I can ask you to turn your microphones on as well, please. Thank you. So what we would like to do is invite you to take part in the Q&A session. So thank you again to all the panelists. Your talks today have really clarified interesting opportunities and trends in the cancer-related pharmaceutical business, and it's provided an understanding of quite interesting development opportunities as well. So following on from that, we'll begin asking our experts some deeper questions on some of the subject matter that you discussed earlier, and hope to respond to some of the questions that have been submitted in chat as well. I have a couple of questions which I'd like to start off with if possible. And by all means, please do answer. It's open to all of you. We do invite conversation. So, you know, if one person wants to answer and if another wants to add to that, or perhaps wants to have a different sort of response opposing that, then that's absolutely fine as well. So to kick off, I'm quite interested from the perspective of technology, what technologies actually caused such a rapid vaccine development, so such rapid cancer vaccine development. It'd be interesting to hear about those technologies and what actually drove that as well.
Yeah, I think there have been a couple of developments, which I also kind of highlighted, I think, in my presentation. So I think firstly, people started to realize that cancer vaccines on their own are probably not efficient enough to actually treat the cancer in the majority of cases. So you will need to be think about combining the cancer vaccine with something else. Well, we didn't have a lot available to us, of course, in the early 2000s. So now we have the checkpoint blockers, and they form a very nice combination therapy there. But the other thing is that we really start to appreciate that that you really need to get a certain quantity of T cells and also quality of T cells to actually drive an anti-tumor response. So quantity meaning, if you, for example, look at some of the antiviral vaccines and the COVID vaccines, and if you look in patients there, actually a quite substantial amount of circulating T cells of these patients are directed against the COVID vaccine. So very strong immune response. And you don't see that a lot in the cancer vaccines. So you really need to have an immune response where at least more than 1% of circulating T cells is directed against the antigen of the cancer vaccine. And that T cell should be both a CD4 and a CD8 T cell. That's very important because then you actually get the most efficient CD8 T cell activation and also the memory response. The memory response is also a very important one. So I think those are probably key points. And of course, now we also have the technology, right, to kind of identify those patients that can actually respond to the treatment. We have the cost of DNA sequencing, RNA sequencing have come down quite a bit. So now you can start to use all kinds of parameters to select patients.
Would anybody like to add to those points? Sorry, Piotr, we can't hear you, sorry. I can see you're unmuted. Yes, there you are.
I may comment this question. I think science is the reason for this. Currently, we have a big direction in science known as the omic sciences. And omic sciences can be considered as a high throughput technologies. And they appeared in science many years ago. And after that, we can able to analyze numerous molecules in single run. And this is very important. For example, we can take new antigens just to find genes, highly mutated genes. We should apply genomics technologies. This is very important. It means that technologies we usually use in science, we are going to apply in biotech. If we have genes highly mutated, we should know that we have mRNA. It means that we should apply transcriptomics. But if we have mRNA, it doesn't mean that we have protein expressed from a gene. This is a big problem. There is not correlation between mRNA and protein. So to find protein, we should apply proteomics. It means that we should apply high throughput technologies. And that is why I see that we have a rise of technologies connected with anti-cancer vaccination. All the proteomics can be used to find tumor-associated antigens in high throughput way. Moreover, you can realize from my presentation, Antigenic Essence technology is based on proteomics. Proteomics is high throughput analysis of proteins. So we're able to generate a big data in different levels, and we can analyze them by applying bioinformatic treatment. For example, artificial intelligence can be applied. Now, that is why I think all is about science, and what we use in science, it will be applied in biotech just several years later. That's all. Thank you.
So just following on from that as well, and I know this was touched upon earlier, with regards to the technological challenges, why did so many trials fail in the past?
Okay, maybe I can comment this question. Because we haven't principal solutions. If we are going to use tumor-associated antigens, it's definitely we will deal with tumor escape. And I don't see solution for this. CAR-T technology can solve some problems connected with MHC restriction. That's all. That is why CAR-T succeeded, but only for particular cancers, only for particular. And I think this is a rare exception than rule. CAR-T technology doesn't work for solid tumors. I think we have same situation with new antigens. We can improve different parts connected with development of vaccine based on new antigens. But I don't see a strong base to succeed in this direction, because trying to use highly mutated antigens in order to produce stable vaccine, very questionable. I don't think so. We know that tumor is heterogenic, and if you take a sample from tumor to prepare vaccine based on new antigens, it doesn't mean that if you take sample from other side of tumor, vaccine will be same. Only if tumor is one clone generated from one clone of tumor cells, you may expect that new antigen vaccine can work. In other case, you will destroy a part of tumor, and you hope that this effect will be evident. That's all. But other part of tumor will be able to grow more rapidly. The empty place will be occupied by other cells. So there are no principal solution for this. What we can do? We should have a very simple and very straight approach to solve main problems. For example, Antigenic Essence technology may be considered as such approach. I think we proposed a very good solution and principal solution based on main principle of vaccination and removing all shortcomings.
Okay. So just putting this back out onto the floor, how would that relate to, and I think one of the questions we had earlier asks about this as well, to the more rare cancers? And sort of spinning off from that question, I believe it was asked about whether we should be looking at personalizing these vaccines for those that are affected by the more rare. I'll just double check who I think asked that question. I think it might have been Robin Privet. For the rarest tumor types, I would think you absolutely need to personalize vaccine as well. So while your response, Piotr, makes complete sense, how would that relate directly to those lesser known types that aren't as easy to combat with those methods?
Oh, pardon, can you repeat the question?
So with what you've said, we talked about the challenges, and we talked about the failures in the past. And it seems it's a great, your response was sort of, you know, one rule for implementation overall for the rollout and development of vaccines. But how would that work with the rare vaccines? How would you engineer that, and how would you be able to tailor? And I think one of the comments from Robin Privet earlier on was that for the rarest types, you need to personalize your vaccines. And when you're talking about personalization, the kind of mass formula doesn't necessarily apply.
Okay.
And that's open to everybody as well.
Okay, I can answer this question. I think if we have very straight approach, we shouldn't deal with personalized vaccination at all. Maybe it's my wrong, and I didn't clarify it in my presentation. I said that we are going to use primary culture, and someone can think that this is a personalized approach. No, this is not a personalized approach. Primary culture relates to one batch of our product. We should use primary culture in order to prepare vaccine suitable for targeting of microvasculature and tumor vessels. We can read cell surface. That is why we know we can't use immortalized cells. That's all. That is why we're going to use primary culture, and we believe that we will succeed. That is why we are going to vaccinate numerous people. We are going to produce a relatively cheap the vaccine. So personalized approach is very complex, very expensive, and mainly relate to new antigens. This is not our approach. Maybe in future we can produce. We're able to do this. Our technology suits for this, but I think we should start with vaccine which can be produced for all people, and maybe in far future, personal approach will be also applied. So my personal opinion, we shouldn't make many efforts in direction of personal vaccines.
So
and new antigens too.
Of course.
Sorry.
So one of the comments actually from an attendee who remained anonymous was that his daughter, his or her daughter, apologies, was actually getting a personalized vaccine due in December to keep her in remission, and it has taken 6 months to analyze and produce. So obviously, from what you've said, there is a case for it, but it's a longer process that, you know, a lot of...
Okay, just about time. Just information about time. To produce, for example, our Antigenic Essence, 3 months is required. But if we're going to about personalization, we have personalization in case of dendritic cell-based vaccines. We are going to use allogenic antigens, but dendritic cells are going to use autologic. And in this case, if we have already prepared antigens, we can produce dendritic cell-based vaccine within 1 week, because we should just isolate dendritic cells and load with antigens, and some portion will be stored, but after 1 week we can already vaccinate.
So there's a scale. Thank you, Piotr. Dr. Goches, would you like to weigh in on that as well? Or
I think, you know, it's an interesting question, of course, if you can go for if you can make a personalized vaccine for a rare tumor. There's not a full answer to that. I mean, it really depends on the tumor. If it's a tumor that has a very low amount of new antigens, then it's very difficult to make a personalized vaccine, right? So then you need to come with a vaccine targeting, for example, a tumor-associated antigen or a cocktail of that. And also,
let's use, for example, like a I'm trying to think of, say, SRCC or something similar along those lines. Does that apply to this case here?
SI?
I think it's, just as an example, I think it was signet ring cell carcinoma as an example. It's quite rare, difficult to detect, but very, very unique to the individual.
Yeah, it's hard to say. I mean, you really have to sequence the tumors and see if you have a suitable number of new antigens in there. And then, like Peter explained, you have to be able to predict which of those are actually immunogenic, are presented on the surface of an APC, and also that's also processed in a correct way by MHC class molecules. And then you actually have to make a product, infuse it back to the patient, and probably you need many new antigens. So if you don't have that in the tumor, then the neoantigen approach won't work. So then you need to start to think about different approaches, such as the tumor-associated antigens that were also covered. That can still work, of course. But for the vaccine to work, you need to also think about, activating the APC, right, with an adjuvant, or combining with PD-1. That really depends also on the tumor microenvironment status, right? If there are no T-cell infiltrates in there, it's going to be difficult to get a response with PD-1. Having said that, for example, glioblastoma, that's a very poor outcome brain tumor. That's considered a cold tumor, but for some reason, people have been able to actually trigger immunogenicity with tumor-associated vaccines in that tumor. That was quite a surprise to the field, because these tumors don't contain any T cells, or very hardly any T cells. So sometimes cancers have surprises there. It's really you really have to look then in the patient sample. So sequence it, and then you can get a sense of it works or not, if it will be suitable for a cancer vaccine.
I think there's a common theme with some of the questions and comments that we've had in chat is, obviously a lot of people would like the idea of personalization, but appreciate that you can't necessarily mass produce that. And it might not be as effective in terms of the time scale to be able to make that much of an impact. So just moving on to some of the questions in the Q&A, I feel as though I have to respond or ask a question posed by Jose Sanchez, who should have really been on our panel. You've got some very, very interesting questions there. So we can choose, perhaps, if we look at one of your earlier questions. So could anti-cancer viruses be replaced by CRISPR CRISPR vaccines, and can cancer cells be mutated back to healthy cells?
No comments.
Is that quite a specific question?
This is related to editing of genome, how it is connected with cancer vaccine. Maybe, but I don't know.
No problem. Well, perhaps we should go back to the concept of technology. So another question from Jose is, AI pattern recognition should have output neurons that recognize patterns, but also it must have an additional output neuron that indicates probability of unrecognized pattern for a certain region. Does the AI have that neuron? Such output neuron leaves a door to spot new patterns instead of delivering bogus output. It's a question for researchers to the AI company. I suppose it's posing a thought, but also asking the question, whereas whether AI has that neuron. Natalia, would you like to, is that something that you could comment on?
Yeah, of course. Could you please repeat the question?
So it's a comment and a question. AI pattern recognition should have output neurons that recognize patterns, but also it must have an additional output neuron that indicates probability of unrecognized pattern for a certain region. Does the AI have that neuron?
Yeah, I think that this is definitely the Christopher's area. So just to make sure that this question is answered properly, I would give him the spot right now.
Christopher, would you like to take a stab at that?
Sure. I definitely think there's AIs that are in development that if you are scanning a wide set of known protocols and profiles in a particular sample set, if there's something that's outlying, you can certainly have it focus on what's left over as opposed to just what you're looking for on those known ones, so that you'll have what's unrecognized and then be able to work on it from that other end. But you'd have to exclude the items that you know are already existing before you can today find the things that are the outliers. So you have to step into it kind of from a backwards scenario.
Okay, I may say several words about neural networks and the application in biotech and in science. I think artificial intelligence can be easily applied for some fields of science, of biotech. For example, to find new composition of drugs, mainly low molecular compositions. And we know such approach known as QSAR. It means quantitative structure activity relationship. And in this area, artificial intelligence can be applied with big success. Currently, it's very difficult to apply artificial intelligence to big data. This is a big problem. We can see a huge number of scientific papers because we have numerous bioinformatics and we have numerous databases with freely available big data related to different type of data, proteins, low molecular substances, genes, and we have a numerous application of artificial intelligence, but we will not see a very successful examples in industry because application of artificial intelligence to big data, to real big data, make situation more complex. Things become worse. But maybe 10 years later, I think this will be a real trend. Now this is a real trend in low molecular design of low molecular drugs and in science. But 10 years later, we will see how artificial intelligence will be applied in the industry.
Perhaps even sooner. A question for Natalia and potentially Andre as well, referring to how cancer vaccines overcome the challenges that tumor antigens rapidly mutate to evade the immune response. So just to repeat that, how will cancer vaccines overcome the challenge that tumor antigens rapidly mutate to evade the immune response?
Yeah, I think I can start with this question. So first of all, as we discussed, one of the main approaches is using multiple antigens in one cancer vaccine. So if, for example, we're having just few neoantigens in constant, it means that if we are constantly targeting only these few neoantigens, at some moment of time, there will be some cancer cells group which will develop the mutation and start a new clone or like a new tumor which is resistant to this treatment. But if we're targeting multiple, like dozens or hundreds of antigens at the same time, tumor doesn't really have this chance to escape that easily. And at the same time, also we might target not only cancer cells which are mutating really, really fast, but vasculature of the tumor, like microenvironment. And by microenvironment, I mean the environment around the cancer cells, which includes vessels, fibroblasts, signaling molecules, and other factors. So I would say that these two are main approaches how we avoid this tumor escape because of the rapid mutation rate.
Thank you. Thank you. I
May
think
I add to that?
Natalia is talking about Santavac. We are going to target vasculature and we are going to use the diversity of antigens. This is a way how we can manage mutations in cancer. Thank you very much.
Dr. Gershis, would you like to weigh in on that question as well?
Yeah, I think Natalia was quite right there. I think another approach is more on the clinical development strategy. So the earlier you give the vaccine to patients, so the early in disease process, the higher chances that you have that the tumor's not so heterogeneous. So you have a bigger chance of actually getting good results with your cancer vaccine. So that's why a lot of people are now interested in actually applying cancer vaccines together with PD-1 in the adjuvant setting, because the tumor has been debulked by surgery or chemotherapy. So there are only some residual cancer cells there. So if you then apply the cancer vaccine, then the tumor doesn't have a lot of chance to escape. But of course, then you still need to find a way to actually get your vaccine very early in the treatment line. But the good thing is that most cancer vaccines are actually pretty safe. So that's an advantage.
And what about the political landscape? Obviously, this will vary in various different states and countries. What's involved with that process, with all the various regulators that exist and have their own operating rules? How would that work with obviously a lot of what's been discussed and getting things approved, and particularly when we're talking about vaccines?
Yeah, it's a good question. Obviously, for any vaccine product, you first always have to do your phase 1 trials to show that it's safe. And if you have an excellent safety profile, it definitely helps you to position your products already early in the treatment lines, like in the adjuvant setting. Still, you will be, I guess, a little bit dependent on clinical key opinion leaders trying that, I would say. Of course, also you need to have consent of the regulators. But because normally with a cancer product, indeed, you have to start with very advanced stage patients that progress to later treatment lines. That's always a problem. But if your product is really safe, showing some good immunogenic responses in patients, so a high degree of circulating T cells, good safety there, then you might be able to convince some key opinion leaders to actually try it in combination with PD-1 or a different checkpoint inhibitor. I mean, there are several others emerging now in your target indication. Yeah.
Thank you. And just one of the questions that was asked earlier, and I think we may have answered this as part of your presentations, but just to touch upon it again, in terms of the success rate of such anti-cancer vaccines as of now.
At the moment, it's pretty bad. Unfortunately, I mean, I think that the ProFrench and the BCG bacterial products are probably the only cancer vaccines products are on the market right now. And so far, almost all clinical trials have failed for reasons that there was simply not sufficient immune response in patients, right? Clearly so, there are a lot of companies now in the clinic with their products. So I guess we'll just have to wait for the randomized clinical trials to read out. So it are exciting times, and I think the field is kind of looking forward to get a success there, right? Yeah.
So in terms of those success stories, actually, Dr. Andriy Bavelo, if you're available to answer, what would you say are the most notable success stories in terms of biotech rounds and IPOs recently?
I mean, in the cancer vaccine space, probably first that comes into my mind is BioNTech and Mrna. They have diversified portfolios, but also include some cancer vaccine products, as we have seen in the presentation, in my presentation. So they were both quite successful with their APOs, especially Mrna. And I believe one of the reasons they were so successful is because they were focused on many things, and also the money that they raised basically led them now develop these products as well, and I believe they will get some success. And also, I mean, Sensei Biotherapeutics is the company that raised 133 million, if I'm not mistaken, quite recently, and so they have also the product in the pipeline. But again, the key metrics right now about this industry is not it's not about IPOs so far, because we are in the proof of concept stage right now, and as I have seen on the graph, we will see either very skyrocketing growth, or if many failures occur, we potentially might be quite disillusioned and quite disappointed. But from our analysis, we do believe that due to new technologies coming into play, both on delivery side and on the antigen selection side, we do see some hope that next year, maybe in the next 3 years, we will see some preclinical success stories and probably clinical success stories. And of course, going from 0 to 1 will be an important part in this process. And but finally, my last point is that in this game, I would say the most important item for a successful company to have the platform, rather than just a single set. The platform-based business will win here, because in the cancer space, it's important to pivot easily between certain indications, and I would say the platform-based businesses here will certainly be heads ahead, like Mrna, for example, or Antigenic Essence, or some other technologies like that, because they can simply pivot quite easily, relatively easily, into certain cancer types, certain patient subgroups, etc. So that's important.
And finally, I think, apologies, I know we've run over, but if I may just ask one last question, which can be put out there, and I think, Dr. Birches, you may be first to answer this as well, which is the critical bottlenecks for startups, so biotech startups. What are the critical bottlenecks that they would be facing, especially in the initial phases of company development? And what advice you would have? I think we do have people that have joined us today who could be from such companies.
Yeah.
This is out to the floor.
Yeah, so it's definitely not an easy time for a cancer vaccine developer to be in, for reasons that the presenters have shown. There's a lot of competition, people using different types of antigens, different types of platforms. So it makes it very hard for investors and also pharma to evaluate those propositions. And also immuno-oncology in general is a very competitive space. Cancer vaccines are probably a very nice combination partner with anti-PD-1, but almost everyone in the world is trying to combine their drug with anti-PD-1, and that does not necessarily have to be a cancer vaccine. So you're not only competing against the cancer vaccines, but you're also competing against different treatments. And in the end, what counts is, of course, that you are able to transform the standard of care of a certain target indication. So I think as a company, what's very important there is that you start to look into the indications where your products would fit very well, make a couple of target product profiles, and where you highlight kind of where you're going to, what will the product label of your cancer vaccine, what will be the combination product, how will you select the patients, those kinds of things. And be prepared that the landscape will shift a little bit over time when you ultimately get to the market. I think you need to be preparing a pretty strong preclinical data package these days. So some of the things that are already presented, so showing basically all the hallmarks, the cancer immunity cycle in preclinical animal models, good efficacy there, and the memory responses is very important, I think. And maybe as a nice to have to actually bring some human ex vivo data there as well. I don't see a lot of companies doing that, but it definitely de-risks the company from my perspective. Because, yeah, I mean, in the end, animal models are not so predictive for the humans. So there's only some, I mean, they're still useful, but not as useful as you wish them they would be. So if you have a combination of both, that definitely helps. Yeah, so just spend a lot of time there to make a very strong preclinical package, be very rigorous, and carefully look also in the literature what other people are getting in terms of immune responses and efficacy, so you know where the bar is that you have to cross.
Thank you. Natalia, I see that you just wanted to quickly comment on what Jose, I believe his name is Jose Luna, not Jose Sanchez. I stand corrected, apologies. Just to respond to his question, his initial one, which was virus, I believe, is it viruses against cancer sounds compelling. If they mutate, they could attack healthy cells too. What measures are there to prevent such viruses going rogue?
Yeah, generally these questions are really interesting, and I would say that viruses which are programmed to attack cancer cells usually face another problem, and this problem is quite opposite to the one described here. Basically, when oncolytic viruses are injected into the human body, the immune response develops too drastically, which means that human body produces too many neutralizing antibodies, and these neutralizing antibodies deactivate oncolytic viruses too fast. That is why generally the action time of these oncolytic viruses is too short, and basically researchers are trying to make it longer and at the same time of optimal efficacy. That is why no, I wouldn't say that oncolytic viruses are likely to mutate in a way to attack healthy cells, because all the genes are deleted from their genome, which could be responsible for targeting healthy cells. But for instance, these oncolytic viruses are very likely to be deactivated by neutralizing antibodies. And the dilemma here is that since we're using the activity of immune cells, of human body, we cannot really suppress the immune response. So we should keep it active, but yeah, at the same time, it's a really, really fragile balance.
Okay, thank you. Are there any final remarks from our panelists before we close today's conference?
I think there was one remark about the cost of personalized cancer vaccines. Yeah, I mean, clearly so the cost of goods are more expensive than an off-the-shelf therapy. Mm-hmm. But it's also, of course, very much dependent on the vector that you use. I think the peptides are probably going to be very expensive, maybe too prohibitively expensive, whereas DNA is something that is pretty cheap. But of course, you still have the cost associated with actually sequencing the tumors, although those costs have come down. But you still need to do the analysis and actually make the products. But I do think, you know, if you get very profound clinical responses, then you know, of the type that people have seen with CARs, for example, or the Lulux CARs, which are also very expensive to make, that there is a room, that there is a path forward. But then, of course, the responses need to be truly stellar.
Mm-hmm. Any other final remarks?
If we can go back to the question regarding CRISPR, I remember we skipped it, but yeah, I just can give a fast remark. That, like, first of all, cancer cells are not likely to mutate back to healthy cells. It's pretty much impossible because their genome and epigenome are highly aberrated. And also regarding whether CRISPR could replace anti-cancer viruses, well, I would say that CRISPR can be efficiently used to fix some of the overexpression problems, for example, or some mutations, but at the same time, we should be aware that even though CRISPR is quite a precise genome editing tool, it still has lots of off-target activity. So it was discovered just like with the whole genome sequencing technology, that basically if we are using CRISPR, we are not targeting often only our target gene. That is why, because of these off-target activities, it still remains a concern and it still requires more research before it can be actively implemented into the clinic, even though CRISPR is used for CAR-T technology, but that's a little bit different.
Thank you very much. So we will now be, if you just bear with me, wrapping. I don't believe there's any other questions. I think, well, there are several other questions and I'm sure we will find a way to address these individually. Okay, so just want to say a big thank you to all of our speakers for the panel discussions and for your talks as well, and also for the several interesting questions and comments from our audience. So thanks again for joining us today for the discussions. We really do look forward to seeing you all in the future for upcoming events. We will be delivering an updated version of this conference website where the talks of all of the speakers today and the panel discussions will be stored, so you can refresh the event anytime and share it with your friends and colleagues in the field, which is obviously helpful for any of the presentations that were missed, or if there was any technical issues, which we do apologize for, that content will be made available to you. I would also like to thank everyone who tuned in to today and to ask you, please do sign up for future events, updates and announcements at our website, which is www.deep-pharma.tech. And please do stay tuned for more analytical reports and IT platform releases, announcements and updates on the subject of advanced technologies in the pharmaceutical industry and investments in this area as well. And for now, you may check out very new reports that have been published on the website. And one of them, as mentioned earlier, the 5 high-impact cancer vaccine platforms. Please do see our contact information and links to the launch reports once again. I thank you all for attending and we hope to see you soon. Please stay safe and take care and have a good evening or morning or afternoon, depending where you are.
Thank you all. It's very interesting to be part of this panel, so thanks.